Good morning, everyone. I'm Ajay Pillai. I'm a program director at NHGRI. Albert Lee, who's a colleague at NHLBI, and I will give the first presentation, which is essentially a welcome. We have some very interesting news. We have 600 people who have registered for this meeting. That is a phenomenal number. It either reflects that people just register or that people actually are very interested in coming and listening to us. I also suspect that people will not sit for the entire 2 days of meeting. They'll come in and go out and stuff through the different sessions. So anyway, so starting off, let's see. I'm going to try and share my screen. And so hopefully all of you can share my screen. Like I said, this is just a welcome presentation. Tell you a little bit about what we are going to do today and thank a whole bunch of people. So LINCS is a common fund program, and it essentially started. Common fund programs have usually phase 1 and 2. Phase 1 is like a pilot thing, and we started this in 2010, and that was in the hopes of trying to do systematic perturbational biology using high quality, large scale assay readouts while minimizing costs in complex human cellular models. That's the entire aim of LINCS. Create data sets that people can utilize for addressing many biological questions. The screen, the slide in front of you shows LINCS essentially by numbers. We have many, many groups all around the country, part of the LINCS consortium. We study different types of perturbations in many, many different cell types, mostly cell lines of one kind or another. And we undertake many, many different types of assays. And all of this data, as you will see throughout today, data and visualization tools that the LINCS groups have made available. Of course, such a big enterprise over so many years wouldn't have succeeded without the hard work of a lot of people, a lot of postdocs and staff scientists, and of course, our PIs who lead the dance throughout the past years. And essentially, what we found that doing perturbation biology at scale and reproducibly continues to be hard, continues to be challenging. Along the way, we have overcome a number of challenges, and you will hear some of them today. As I sort of indicated, LINCS was aiming to produce a community resource, and so data release and the associated metadata was very important for us. And we've spent a large amount of our internal resources making sure our data is right and our metadata is good and appropriate and useful. And some of that has paid out, as you will see in some of the presentations, as well as some of the results that we have from counting up the number of publications where people have used LINCS data. The consortium itself has generated over 200 publications in over 30 different analytical and visualization tools, and tens of thousands of users have used the tools on the LINCS website. We have also seen hundreds of publications by groups outside of a consortium that have used LINCS data for their own purposes to look at diseases that span nearly half of NIH institutes. That means it spans a very wide range of human biology and disease. So effectively, LINCS's impact has been much broader than some of us had initially anticipated. we were also very pleasantly surprised by the number of companies that have actually used links data and have taken approaches that have been pioneered and by links within their internal scientific and drug discovery efforts. And you'll also hear a little bit about what the FDA is trying to do in using links types methods for their regulatory purposes. So this is a slide. I mean, there are many ways in which we can look at how links is useful. Albert and I have always found this fascinating thing, probably because it's primarily restricted to something that we can do inside NIH. So this is counting up the number of applications that use links data and propose for grants. And what we have found is a steady growth of the number of applications. If you look at this curve, you know, in 2020, within just the first receipt cycle date of applications, there's been more applications that use links than the entire year in the past 3 years. So we are not only growing, we seem to be growing sharply. But some of this may be related to, you know, COVID-related things, but we'll see. And the vast majority of these applications, of course, are not from Links Consortium members. And the other interesting thing that we've been hearing is that we've been hearing links mentioned in study sections. So people look at applications and say, hey, why didn't you use links data to do some of your preliminary analysis, which is great recommendation. So I'll hand over the baton to Albert. Albert, take it away. We can't hear you, Albert. You're muted.
Sorry. I'm just going to give a quick snapshot about what we're going to cover in the next couple of days. So, you know, Ajay described a large data and analytics resource that we've created by links. But instead of talking about the details of the resource, we want to talk about what impact and what we've been able to learn, because that's ultimately what Link was built for, is to try and have a resource in the community for people to make new discoveries. So in the first day, in the first session, we're going to hear primarily from our own links investigators and hear about what new biology they've had. In the session 2, we're actually going to turn it around. We're going to hear from people from outside the Links Consortium, people that have been using our data. And frankly, it's been really fascinating to hear about the different ways they've used it and what they've learned and how they've applied it. And there's so many examples, I think, that are really cool. We've actually spread it out into 2 sessions. So we'll hear about some in session 2 today, as well as in session 5 tomorrow. We'll wrap up the first day with session 3 by some hands-on parallel workshops, as well as about a dozen poster sessions. In the workshops, you'll get to hear more details. You'll get to kind of roll up your sleeves and learn how to actually access and analyze data. And in the poster session, you'll hear about some of the cutting edge research that our own links investigators are doing right now. Tomorrow, we'll start off in session 4. You know, up until that point, you'll have heard primarily about single assay analysis and what people have learned from single assays. But Links generates many, many assays. And in session 4, you'll hear from our investigators about how to do integrative analysis across multiple assays and what additional new insights can be gained by looking at integrative analysis. Again, session 5, we'll hear more interesting vignettes from the research community outside. And session 4 will end the symposium with additional new different workshops and different posters. Ajay, let's go to the next slide. These are just some quick acknowledgements. I'm not going to go through all these names, but obviously there's a lot of people that have helped us make Links the success that it has been. You know, the Common Fund, obviously, we really appreciate their support, as well as the program leaders in the Common Fund that have really been helping us stay in the lane. Our co-chairs are the big heavy hitters of the program. You know, when we need to spend a lot of money or make a major decision or major change, these are the people that say yes or no. And we really appreciate their support and enthusiasm throughout the program. There's a lot of peers and colleagues of Ajay and I at the NIH on the project team. And these people in particular, I think, have been really helpful, steadfast, and very supportive. We've had a number of external program consultants. These are people that outside the NIH that are advising us on the LINKS program. And again, we really appreciate their insight. And I think many of them have been with us for a long time and have really rolled up the sleeves and looked under the hood and asked, I think, really important questions, but really made the program much better. There's many people in the past that have been involved with LINKS, but I think they really made a big impression for us. And of course, the symposium organizers, many on the LINKS team that have helped to shape what you're going to hear about in the next couple of days. And I think some of you have heard from our Greg Richards already, our subcontractor who's handling meeting logistics. And Greg will take good care of us if we have any problems throughout the day. And at the top in the middle there, I just want to mention the program analyst. For all of you that have organized conferences and workshops, you know that there's always one person behind the scenes. They don't get a lot of credit, but they're the ones that sweat the most, stress out the most, and are on top of every detail. And for this symposium, it was really Julie Kim who was that person for us. I'd really give a special shout out to Julie. Thanks very much, Julie, for making this a success. Okay, so that's all I wanted to say. I think I'll turn it over to Greg if there's any logistical announcements, and Steve Finkbeiner, who's our moderator for the first session.
Sure, just a real quick announcement. So during the sessions, attendees will be able to submit Q&A using the Q&A button at the bottom of your screen. At the end of each of the first two sessions today, we will have a dedicated Q&A session. So during each of the presentations leading up to those sessions, feel free to submit any questions you have, and then we will try to respond to as many of those as we can during the Q&A period. If anyone has any questions, feel free to shoot them my way at Greg Richards, RLA.
Great. Well, my name is Steve Finkbeiner, and I'm pleased to moderate this first session, Undertaking Large-Scale Perturbation Studies, New Biology and Lessons. We're going to hear the first talk from Peter Sorger, Generating and Mining Signatures of Drug-Induced Perturbations. And I just want to reiterate Greg's advice. Please, as a question occurs to you while you're listening, if you can enter it into the Q&A box, that would be great. What I'll do at the end during the 30-minute Q&A session is hopefully gather those together and then try to go through them. So thank you very much. Peter?
Peter, I think you're muted. Oh, so when you're sharing your screen, move your mouse to the very top middle of your screen. You should see a little green bar. When you hover your mouse over it, you should see at the controls. There you go.
Okay, we're seeing this correctly now?
Looks great.
Great, thank you very much. It's a great pleasure to be able to kick off this meeting. I have a few relationships to disclose, none of which are particularly relevant to what I'm going to comment on today. But I want to start off by really thanking Albert and Ajay, who've just given the introductory remarks. Albert's a man of mystery, very hard to find a picture of him, but I did grab one off the screen a few seconds ago. They've had to live through, I think, all of us trying to make our way through a sort of subtle and complex set of experiments. And I have to say, particularly my relationship with Ajay over the last few years has been one of the greatest pleasures, actually, of my scientific career. So thanks to them for making this project what it has been. So in our center, the goal has been to take perturbations, not surprisingly, put them onto cell-based assays, do molecular profiling, and derive signatures. And the particular perturbations that we've been interested in are kinase inhibitors primarily. That's the most extensively studied class of drugs. About a third of all of the roughly 540 human kinases have relatively selective and high affinity inhibitors, and they represent the majority of targeted therapies in oncology and increasingly other diseases. We also looked at cytokines, growth factors, and as a drug class that is the other end, just beginning to be developed, we looked at DUB inhibitors and epigenome modifiers. Pretty diverse set of cell lines, primarily immortalized lines, but also cardiomyocytes and neurons. We'll touch on that briefly. Molecular profiling was done in a kind of multi-omic approach. And then a variety of signatures that I'll talk about, but so will Caitlin Mills in the session tomorrow. But as we went forward, we realized that actually there were additional aspects of this that were critical to understanding signatures, and that is phenotypes. And so you actually want to know if there is a molecular change in a cell type as a consequence of perturbation, is that actually consequential with respect to some key biological variable, which could be growth or motility or cell death. And of course, we want to interpret these signatures in light of what we increasingly know about molecular networks. And signatures can both inform molecular networks, but networks can also provide input into signature generation via feature selection. Again, something that Caitlin will talk about tomorrow. So the links approach, although seemingly simple, actually has these additional features, and this type of integration remains an ongoing task. And in many cases, the initial sort of statistical approaches we used were now replaced by machine learning, but many of the same fundamental challenges remain. A critical thing in our work, as in all others, as you will see, is that there's the systematic collection of n-dimensional data cubes. So trying to fill in as much of a data cube as possible. And the type of data that I'll show will often have a response matrix. If you just zoomed in on a piece of that, you'll see that there are many features at multiple doses and multiple drugs in this case. You then go into one drug. You'll see a single dose response curve, each of which is associated here with a measurement, for example, of cell cycle progression. So in our work, a typical data point would be 8 to 10 dimensional with respect to dose time and the measured features. And that actually represents a type of data that's common in sort of low throughput science, but actually the statistical methods for analyzing this type of data in volume are only now being developed, and we expect the type of data that Links collected to continue to be valuable in this regard. So the HMS Link Center was obviously, like other link centers, primarily charged with resource generation. And I'm going to show you briefly the 4 types of resources that we developed. The first of them are perturbagen libraries and their annotation. The second, and by far the richest from our center, is analytical tools, and those are both experimental and computational. Response data and signatures, and we've released over 350 data sets. And then specific use cases, so ways in which the tools, for example, and data could be combined in order to make a scientific discovery. And those we found in general have driven interest in both the tools and in the underlying data. So let me just start off quickly with just one little example, a brief vignette of an example, essentially here, an exemplar. And here we're looking at actually 4 kinase inhibitors for recently developed within the last 2 years for HR+, HER2- advanced or metastatic breast cancer. This collaboration involved a clinician, Dan Urich, and 3 key Links members here. You'll hear from Mark Hoffner, I think, later. He's now at Genentech. Caitlin will be presenting, and Kartik Subramanian, who's now at BMS in Boston. And these drugs are actually all marketed as being effectively identical. And they were exciting when introduced because they were able to block the G1S transition via inhibiting specifically CDK4/6, and they therefore had an anti-proliferative effect. So the Links approach to getting at sort of underlying mechanism of action in this case was to perturb the cells. So here's, you can see, get a sense of the matrix at the top here. We have 3 different drugs, 3 doses, 2 times, 6 cell lines. We collect, in this case, an RNA signature, and we go to the connectivity map that you're going to hear about from Todd. We mine signatures from those connectivities. So this is sort of the enrichment score. Each signature in this case is a ranked ordered gene list. We can then take different signatures, plot them now in a signature discovery phase. So we have a pan-CDK signature and a pan-CDK4/6 signature. And you can immediately see in this signature space, the different drugs actually plot quite different locations, as shown by the different colors of the dots. And this was eventually explained when we got as far as individual targets. And I'll give you one more slide, and Caitlin will tell you more about this tomorrow. So the goal of these drugs was to inhibit 4/6, not to inhibit CDK9, which was part of an older generation of drugs that did not find clinical use, alvocitinib, because they were too toxic. And you can see to the first approximation that ribo and palbo are fairly similar to each other, as in fact advertising at AACR would suggest. But abemaciclib is actually quite a different drug. And this had been hitherto masked in the literature. It's active against the primary cell cycle regulator CDK2. That was found through Blink's discovery. It was a controversial idea at the time, but in fact, Pfizer has gone on now to develop a drug with explicit these properties in order to increase efficacy. And our clinical collaborators have a trial underway to show that patients who are non-responsive to ribo and palbo would be responsive to abemaciclib. Now, another key to success that I'm not going to talk about as the focus of Caitlin's talk is that actually we didn't find most of the necessary information from any single assay. We really integrated, in this case, 4 data types: biochemistry, microscopy, sequencing, and mass spec. And I show pictures of the key staff who were involved. This led to this sort of exemplar publication. And I sort of analogized this to looking at different parts of the elephant. And actually quite explicitly, if I had the time to go into this, you would see that different analytical methods actually focused on different aspects of mechanism of action. And together, they gave us, I think, what is the full picture. So I think one of the surprising things then that's been critical to our thinking about perturbational libraries is that even when you have clinical grade compounds, the target space is remarkably poorly annotated. And in many cases, drugs are known by a single target. We call that their nominal target. And signature analysis shows that may not even be the primary efficacy target. And so Ninka Morit here has developed now the small molecule suite, one of our links tools. And this allows you to mine. It's a completely automated, data-driven approach to finding drugs and target combinations. And it's now automatically updated based on data in Kemble. And it provides a much more sort of sophisticated look. For example, you can ask questions: if I have a protein and I want to find the best perturbation, what is it? Or if I have a perturbation, what proteins will it inhibit? Here's nilotinib, for example, and I can ask how similar are any other compounds that have been released into the public domain, and is nilotinib, in fact, a good drug for inhibiting a particular set, in this case of EGFR type tyrosine kinases. We've also updated what we know about kinases. You would think this was all sort of established 20 years ago, but as the genome has progressed, we've come to understand target space in a much more sophisticated way. So perturbations and targets need to be understood together. And in work that now continues in the investigating the druggable genome, we actually now are fully annotating these target spaces, again, with automated discovery tools that allow them to maintain up to date without continuous human intervention. Now, the fundamental measure of the relationship between a perturbation and a phenotype is dose. And the way we think about that, in the case of a drug, for example, is the better drug is the more potent one, and that would be the blue curve here. But of course, these kind of sigmoidal curves can either vary at their midpoint, which would be called IC50, in how effective they are at the maximum dose Emax, or in the slope. And at the time we did this about 5 years ago, we actually went, it hasn't changed fundamentally, but we went into PubMed and we asked, what do people focus on? And the answer is entirely IC50, so affinity. And this led to really one of the first sort of, I think, discoveries in our center and a set of tools. And that is to go back to all of the large-scale dose response data, reparameterize it. So you can imagine a FINT here where we look at these different variables. And what we discovered was, in fact, in real dose response data, all of these variables are equivalently contributing to total difference in perturbational activity. And here you just see two sets of lines. You can see very different slopes for a TORC inhibitor and a PI3 kinase inhibitor, even though we think of those as successive molecules in a related set of pathways. And so this led us to a whole series, a whole sort of branch of links that we never imagined, which is actually taking this idea that's over 100 years old and trying to rework it in terms of modern data science. It led to the development of GR metrics. This is a new way of computing dose response, and what we do is correct for differences in cell division. We found that existing preclinical pharmacogenetic markers are so confounded that the majority are actually incorrect because of an inability to correct for this effect. These metrics, which are as simple to calculate as what we've all been using, so GR max instead of Emax, also tell us in the case of cell division whether we have different phenotypes between death, cytostasis, and a partial inhibition phenotype. Partial inhibition, interestingly enough, at a single cell level, often is growth of some cells and death of others. Not all cells are partly growing. And this led to a whole series of sort of papers and tools that we put out that have been picked up quite a bit. And you'll hear from Mark Hoffner about how he and his colleagues have used that at Genentech after he left the lab. And also a very productive collaboration with Mario Medvedevic's lab, and Nick Clark is shown here, to build an online calculator that you can use. And we found that uptake of this calculator in industry is nearly universal. Every preclinical pharmacology group we visit now uses GR calculator or something similar. In academe, uptake has been much slower for reasons we don't understand, and we continue to see a lot of papers coming through with this fundamental confounding of cell division and drug sensitivity. And there's going to be an entire workshop on day 2 also run by Caitlin, who's obviously our link star here. Now, it's possible to score a lot in a simple plate-based assay. And this has been, here's just a quick example where we're now imaging cells across, in this case, you can see 4 channels. We can routinely do 6 or more. And these assays have existed for a long time, but they've tended to be fairly cumbersome. So as a consequence, the vast majority of perturbational phenotypes to this day are collected using simple assays like cell titer glow that give you little about phenotype and virtually nothing about cell-to-cell variability. And what Caitlin has developed is a fast, cheap, reproducible assay, $1 per condition, that we call DiDrop. It's just being published now. And that allows you to take 384 well plates and for a small increment in cost, actually recover deep single cell data from this. And we presented this in posters, and you'll see one of those later in this session. So here's an example where I think links made it possible for us to push an existing technology and drive the price down by about 10-fold. So now it's really routinely possible to collect this. And there's a clever little trick as to how you do this by using solutions of different densities in order to allow automatic pipetters to collect what would normally be a laborious set of data. The other thing we've used a lot of is DGE version or drug-seq. These are cheap, fast, and reproducible RNA-seq measures. We're now down to below $10 per condition. And this is sort of 3 million reads. As I'll show you in a second, this can be directly linked into the database that Todd Golub has developed at the Broad. Here you can see that these databases also show a dose response. And one of the neat things about working in this kind of RNA signature space, and you'll hear much more from Todd, is that we can now take a query we collect locally for $10. We can query that against the Broad connectivity map. And you can just see, if you look at the diagonals, if we now query with nilotinib that we looked at a moment, we recover nilotinib, but we also cover several other molecules which have interestingly related mechanism of action. So the signature query and generation allows us to rapidly probe for similarities in effect. And this now is deployable at any lab that has a sequencing machine. Now, this has led to the release of some large-scale data cubes, and this has been the work of Mira Chung. To go through, and you'll hear a bit more about this later in the session, just many, many drugs over hundreds of cell lines. And so you begin to get this kind of systematic view that I alluded to before, where the responsive drugs, and you can see the shapes of these curves, is really remarkably different from one condition to the next. And there's been quite a bit of additional work on figuring out what that means. But let me just say in passing, it primarily has to do with cell-to-cell variability that's not genetically determined. We built a database. You can see from this, there are 350 data sets. And this has been the work of a couple of very talented programmers working in our screening center here at Harvard under the supervision of Carolyn Shamu. And this is a place where we can deposit that data, which doesn't fit into the larger DCIC data set. And the complexity here is, of course, you have so many different types of assays as you build these cubes out that systematizing them is only now becoming possible. And I think one of the things we've discovered, to which Ajay alluded, is what we appreciate so much from genomics, the existence of self-consistent, well-annotated, metadata-associated information about cells is available in almost no other space. So proteomics is late to the table. The imaging I showed you has relatively few resources, and the ones that do exist in Europe. So the vast majority of the type of data that we can collect on cell perturbation remains to be systematized, although I think LINCS has now showed us the way forward. Now, one of the things that arose in the course of this work was an issue of reproducibility. There were a series of fairly high-profile papers and then a series of responses to them. It actually got written up in a book about sloppy science called Rigor Mortis, about the idea that when different centers measure drug response, really something very simple that we all would have thought would be straightforward, from one center to the next, they got different answers. Obviously quite concerning. And so this led to a LINCS reproducibility study that's been presented to Beastie and has been published, if anyone's interested, in which all the centers got together over a 2-year period, collected the same reagents, did the same experiments, and then compared their results. And what I can tell you is the measurements initially varied by over 100-fold from one laboratory to the next. We could get them to vary from one floor to the next of our own department. So if you can't translate an assay from one floor to the next, it's no wonder you can't translate it from a cell line to a human. But the good news is, by the end of this procedure, we come up with a set of fairly simple suggestions that Laura, Heiser, Caitlin, and I helped to put together that would allow others now to get this within a variance of about 10 to 15%. So I think this is a fairly rare example where we don't wring our hands about reproducibility, but we actually build some concrete tools and actually come up with some specific recommendations. Before I sign off, let me just give you one now emerging example of therapeutic discovery directly using signatures. So something that we think of as a pretty exciting, a sort of kind of LINCS application. So what we start off is with mRNA signatures of drug response in neurons, so using this drug-seq method. What we're asking for in this particular project is to say, is there any mechanism of a drug which sufficiently resembles the mechanism of, in this case, Alzheimer's disease, that we have an opportunity for repurposing? We then get mRNA signatures of Alzheimer's disease in patients, and that actually is a large-scale data set that's been collected already by the National Institute of Aging. And then we ask what variables in that Alzheimer data set are related to drug mechanism of action. And that then allows us to pull out a new set of compounds and actually quite unexpected ones that had to do with interferon response pathways that in preclinical models are actually neuroprotective. So I think that's an example where the entire discovery process takes place in this auxiliary space of cellular signatures. Now there is a lot that's remaining here. Among the things we haven't been able to deal with is, I think in a systematic way, is all of the variability that happens from one cell to the next. Here you're seeing a drug-treated cell, and you can see that all the cells behave quite differently. They pulse over time. This gives rise to the bulk signatures that we've interpreted in the current work. And we think this gives rise to some of the macroscopic phenomenon and dose response that I told you about just a moment ago. So what is left for our LINCS program? We have 6 more months to continue in a no-cost expansion. We are now refactoring our website entirely around the following idea: reproducible and reusable perturbagen libraries. We've built 2 LINCS libraries now, and those represent the best kinase libraries as yet instantiated. The analytical tools are, of course, open source. Response data are being integrated with the rest of LINCS. And then these use cases will have individual landing sites on the web so people can see ways in which LINCS data could be applied. So there's a large number of people whose work I have butchered in this short presentation. Here are some of the PIs down at the bottom, but particularly want to thank Carolyn Shamu for all the work she did in organizing LINCS, and Nathaniel Gray for all of his insight into chemical biology and the discovery and invention of new kinase inhibitors. Thank you very much.
Great, thank you, Peter. We're running a little late, so let's move on to the next talk. Transcriptomic signatures for drug adverse event prediction in human iPS-derived cardiomyocytes.
Yeah, this is Ravi. I'm just starting to share my screen.
Thanks, Ravi.
Now.
And just a reminder to anyone who's joining us, please put any questions you have in the Q&A box. We'll get to them at the 30-minute session at the end of this session.
Can you guys see my screen?
We cannot, so make sure that you hit share screen and then that you also select the window.
Oh, yep. Share screen. Hold on 1 second.
Sure thing.
Screen 2, that's why. Okay, can you see it now?
Yes, we can. Looks great.
Okay, sorry, it was screen 2. Okay, so I'm going to talk about what we've been doing at the Drug Toxicity Signature Generation Center, which was, we focused on getting transcriptomic signatures for kinase inhibitor drugs that are already FDA approved. And the drug toxicity we focused on was cardiac heart failure type drug toxicity. As most of you know, a significant number of people who are given kinase inhibitor drugs, the number ranges from, say, anywhere from like 10 to 20%, develop sort of cardiac insufficiency, heart failure-like symptoms as they're on the drug. So even when the cancer is in remission, the cardiac disease picked up. It's a sufficient problem such that almost all big cancer centers now have a cardio-oncology unit that follows the patients as they are on these drug treatment regimens to see what's happening with cardiac function. This was a multi-PI grant. My colleagues who sort of directed this program were Mark Bertwistle and Eric Sobey. Before I forget, at the end, it was a very large team. There's a whole bunch of people who helped us to various parts. And I want to particularly pay sort of recognition to the clinical team because they helped us gather healthy human subject fibroblasts that helped us develop a set of cell lines where we could study this. And this is sort of in the longer term range an important issue because more than half the people who develop this sort of cardiotoxicity to the kinase inhibitor treatment have no prior history of heart disease or cardiac dysfunctions that might account for. Of course, people who have them have a greater propensity to develop them, but there are lots of people who don't who still do this. So the basic approach has been to use transcriptomic signatures, and the question was, could we predict relationship between cardiotoxicity, clinical measures of cardiotoxicity, and these transcriptomic signatures. Initially, in the first 2 years, so we were only in the second phase of the LINKS program. So our center ran for about 6 years, and we are now in this 1 year no-cost extension period. The first 2 years, as we were developing our iPSC cell lines, and I'll get to that in a minute. We purchased some commercials, heart myocyte-like cells from this company called Promo Cells, and tested a range of kinase inhibitor drugs. These cardiomyocyte-like lines, they do come from hearts of adult humans who who were not deceased but passed away for other reasons. But nevertheless, because they have been cultured in vitro, they have fibroblast-like properties, and they actually do not beat and do not show expression of a lot of channels. So I use the term cardiomyocyte-likes to make this sort of clear that while they are derived from cardiomyocytes, they are not quite there yet. But anyway, we tested about 21 protein kinase inhibitors. We used the 3' DGE method that that Peter sort of pointed us towards, and Broad did part of the library preparation, helped for us in these assays. And what we did was we got the transcriptomic signatures and the transcriptomic patterns and asked the questions, could we derive signatures that match with the clinical risk scores as assessed by from clinical databases? And for this, what we did, we have been doing this for a while now for some other projects in the lab. We used the FDA adverse events database to calculate risk scores, and this is now published. We had this paper finally has come out in Nature Communications, so you can look this up. So one can calculate the relative risk for any one of the kinase inhibitors that we tried, and there's a plot in the that you can see, and you can look for greater details in the paper. One of the issues was, you know, with all of the adverse events databases, there's always this question or this issue that we actually don't know the denominators, which is that how many people took the drug. So there's sort of a little bit of a hand-waving type approach where one calculates the relative odd ratios. So one of the questions was FAIRS, the clinical risk scores unique in FAIRS, and how did that compare to other databases around the world? So what Kuhn, Van Hasselt, who did some of this computational work, decided to do was to get the WHO database, which contains data from many countries, including the US, spent time cleaning out the FAIRS part of the WHO database and compared the remainder. And you can see that actually the WHO and the FAIRS for the kinase inhibitors match very well. So we feel reasonably confident that the risk scores we calculate are going to be widely valid across different kinds of ethnicities and populations and so on. So one can take the what Kuhn did was to use sort of a logistic regression type model to fit these things and look at a expression pattern of a set of genes with respect to the predicted and observed risk scores, and he finds pretty good correlation, and you can see the plots. And we had actually, for various reasons, these experiments continued for a number of years. And on the rightmost side were a number of drugs that were tested de novo about 2 years after the initial plots were discovered. And you can see most of the drug except one land on the correlation plot pretty well. There is another aspect to this thing, and there's going to be a poster presented by Reis Rahman tomorrow, which is that the structure of the targets and the structure of the drugs, as well as the structural pattern of the expressed proteins, appear to play a role, so that these kind of transcriptomic signatures can be enhanced by what we are now starting to call structural gene expression patterns. And so there are additional improvements that can be made to make these more specific, and some of this may explain the relative variations I can see. So I encourage you guys to visit Reis's posters if you can. we spent quite about 2.5 years in the beginning, but it's taken a whole time, several years to get this fully done, because of the fact that people who develop these cardiac dysfunctions often are quote unquote healthy, except for the cancer. We decided to create a library of healthy human subject iPSCs that's both diverse with respect to sex and diverse with respect to ethnicities. And we have now finally finished this whole thing, and this paper is now out on bioarchives and being submitted to for a peer-reviewed journal. So we have 40 fully characterized iPSC lines, which include a variety of information. The most important among this, with respect to studying this aspect of drug toxicity signature, is the whole genome sequence. So how does the drug response signature vary from individual to individual? So this is kind of a question we would like to answer using this type of an assay, and which was the driver. Nicole Dubois and Christophe Chenil have a talk tomorrow where they will describe this resource in detail. All these cell lines are well characterized. The data, the genomic data is in dbGaP. The cells are being deposited in Y-cell, so they're just going to be widely available to the academic community as they need to use them. So we had initially wanted to do the experiment on all 40 lines, but given the cost of how things worked out, we were able to do a large number of drugs using 6 lines, and we thought that this would be the initial sort of thing that would allow us to take the whole project to completion to get an understanding. And in doing this, we decided to design these experiments in a way that not only would we have these kinase inhibitor drugs, but we would have other FDA-approved drugs that are cardiac drugs, but have over the years never been associated with causing cardiac dysfunction or leading to heart failure, and see which of these expression pattern signatures varied with respect to the specific phenotype we were expressing. So this is a long list of drugs. The anthracyclines are positive controls in one way, if you want to think about it, because these are all drugs that are known to be quite toxic for in terms of cardiotoxicity. There are a number of cardiac acting perturbagens. And as this list was being compiled, several of our colleagues from the FDA had suggested other drugs for a variety of reasons. They were interested in cardiotoxicity. So we added this list of drugs. All these drugs are FDA approved. So there are known, well-known dosages that are used in humans. So what we decided to do was to back calculate what might be a concentration in the cell-based experiment that would correspond to plasma levels of these drugs after the loading phase, in sort of a steady state phase when the patient is being given the drug. And so we tested it at a single concentration. The questions we were trying to that we try to answer in developing this database or in developing these data sets are: can this be related to a particular cellular functional response? Can this be related to clinical risk? And the last question, which I think would also be very interesting, is can the drug response pattern be related to genomic variants of these cells? So the statistical approaches are relatively straightforward. These are calculated on the basis of log P10 values. So the experiments that we have done for all 6 cell lines for 54 drugs, and developed a large list of DEGs, data sets. And you can see this is the distribution of the differentially expressed genes. these things one can set on choice. And for these initial sets of analysis, we have set an FDR at 10%, which is a permissive level. We can cut it down to say 5% or 3% as we go along to see how much specificity we get at various things. I think the basic question we started to ask, this is sort of a little bit of a meaningless slide if it looks nice and colorful, was that do the DEGs reflect drug-specific responses? And the short answer is actually no. That the initial clustering, simple clustering analysis does not show between these 6 iPSC-derived cardiomyocyte cell lines, 3 of which came from males, 3 of which came from females, and a variety. It does not cluster with respect to drugs in a significant way. However, the responses do cluster quite well with respect to cell lines. And if you see the upper part, the greens, there's one that is sort of more dispersed, but you can see this huge block of green, the light blue, the gray, and so on and so on. So most DEGs cluster by the cell lines, although they don't completely cluster by the drugs. And you can see, although there are some drugs there where you can see the clustering, and we have identified this. So there are some measures for the drug-based clustering that we looked at in terms of the F1 score to see how well this clustering might occur. And as you can see that for a few they do, but for the most of the drugs, we were quite confident that a simple clustering approach is not going to really give us any sort of meaningful results of was there a drug selective response across the different these six different cell lines. So what we decided to do was to, we can ask couple of questions, right? Identify components that are drug specific, identify components that are cell line specific. And there was always this concern, which is why I had worked in these 12 extra drugs, is that some of these expression patterns probably contain a set of genes that are differentially expressed just because of the perturbation, and it is not really related to any specific changes that might be, that the cell might be undergoing, in response to this sort of perturbation. And you can, I mean, Jens calls it the general stress response. That's good. So what we used was we used an SVD-based approach to deconvolute the data into individual components. This is an approach it's been around for a long time. The first paper that sort of showed this was a classic paper by David Botstein and his colleagues in PNAS back in 2000. But the approach actually works well. So SVD can really resolve the cell line specific components with the and the DEG specific components. This is sort of a very recent analysis done by Jens Hansen, and you can see that the cell line specific components he has put in a black box, and you can clearly see that effect. And this is a good, I should go to the next slide. Maybe not. This is a good point to make, because each cell line reflects a different individual, and the cell line specific effect might very well reflect sort of a genomic effect on the drug selective responses. And I have a colleague here, Eric Schatz, who is sort of really one of the pioneers in studying the EQTL effects from the genome in context of disease functions for a very long time. So we've just started to work with Schatz's group to sort of sort of identify the genomic components that might drive this drug-specific responses. Now, we do find DEGs that are drug-specific across the multiple cell lines. So we have eventually now classified over the last few weeks 54 different sort of drug-specific subspecies that allows for clustering by drugs. And this the upper panel shows the F values for this, and the lower panel shows sort of, if you want to call it, the correlation or the relationship between the full data set and this particular eigenassay that shows the value for the drug selective component. So this is kind of where we are. We now are starting to relate this drug-specific spaces across the 6 different cell lines to the clinical risk space. And once we do that and we have a set of eigenassays that specifically relate to the heart failure type clinical risk for KIs, we can start doing 2 different kinds of analysis. One is pathway-based enrichment to understand what pathways from these genes, this set of genes that are in this eigenspace, what pathways are involved and how might they contribute to the phenotype. And secondly, the issue of what these genes and what are the relationship to the genomic variants and the individual cell lines that drive the expression patterns. So we are hoping to get all of these sort of data finished in the last 6-7 months that we have in the no-cost extension period. I think this is the end, and I will stop here and take questions. Oh, no, not questions, but whatever.
Thank you very much, Ravi. That was terrific. Yeah, we'll do questions at the end.
Yeah, sorry.
Running a little late. Our next talk will be the impact of microenvironmental signals on molecular and cellular phenotypes by Laura Heiser.
Thanks, Steve. Do you see my screen now?
You sure do. If you could just put it in present or, yeah, perfect.
There we go. All right, excellent. Thank you so much for the time to speak today and tell you a little bit about one of the projects that we have been working on in LINCS. This is an interesting use case for LINCS in that it is a consortium-wide project that I will tell you about. So I have the great privilege of speaking on behalf of a number of individuals within the consortium who have made major contributions to the project that I will talk about here. The central goal of this project was to really try to understand the regulatory networks that control cell phenotype. If we stop and think about that particular question on the face, seems quite simple, but in fact cells can take on a variety of different phenotypic behaviors. They can proliferate, differentiate, undergo various collective behaviors, migrate, and so on. And some of these processes are fairly well understood, such as proliferation, but many of them are not very well understood in terms of the details of exactly how these processes are controlled at the molecular level. And to date, most studies have been performed by looking at a single axis on which these behaviors may be controlled. So Peter sort of alluded to this in his talk earlier today, sort of thinking about studying just the gene expression programs, which of course are informative, but give just one view of the picture. And what we'd like to argue today is that looking across multiple different assay types and readouts provides a really deep view of various complex phenotypic behaviors and gives some insights into the molecular changes that control them. And so what we did in this particular project was to coordinate across the LINCS consortium to come up with a way of trying to identify the linkages between cell phenotype and molecular state. And as I mentioned earlier, we think this really requires systematically generated large-scale perturbation data sets, and that the capability and expertise really to collect all of these data types is required for comprehensive understanding. And that capability is not something that can be found in one single lab. And so in order to really link across these different types of readouts shown in that schematic at the bottom, what we did was to leverage the LINCS consortium structure and coordinate across the various LINCS data generation and analytical centers to try to come up with a data set that could be used to identify these linkages. And the general approach, which you've heard already, this theme will be repeated over the next 2 days, is to systematically perturb cells and then assess their responses in a variety of ways. In this particular case, what we decided to do was to use extracellular growth factors and cytokines as the perturbation shown there in the first panel. We then are able to systematically measure responses using image-based assays to assess behavioral phenotypes and bulk molecular assays to get a view into the molecular processes associated with responses to these perturbagens. And then to analyze these data in a variety of ways using both statistical and pathway-based approaches, machine learning approaches to try to integrate across those different data types and to gain insights into how the data types are related, how they are related to the phenotypic behavior. And an important thing to note, which also has been mentioned already in these particular talks, is that this is not, I'm listing this here as a sort of one-way feed-forward process, but in fact, the analytics always end up feeding back to help us understand what is the next set of experiments we might want to do, or what is the next set of experiments we might need to do in order to validate some of the associations. And so that theme as well, I will demonstrate in this talk as we proceed. So to get into the details now of exactly what we did was to take MCF10A mammary epithelial cells. This is an incredibly well-studied model system. If you are not familiar with it and interested in this type of study, I encourage you to look into the literature and you will find a number of large-scale studies that have used this model system, which was one of the reasons why we chose to use it, because it is such a well-characterized model. It has near normal genotype. It shows adherent growth, which is a convenient property when running these kinds of assays. It's non-tumorigenic, and so we think provides a reasonable view of what a normal mammary epithelial does. It also shows EGF-dependent growth. This is an important detail. I won't go into all of the details about that particular property. It turns out to be a convenient thing to have a single dial on which one can modulate the growth of a particular cell line. We then identified in a set of pilot experiments a series of ligands that strongly modulate cell phenotype, in particular with an eye toward modulation of proliferation. That's shown in that top panel there. We identified several time points so that we could assess the dynamic responses. And then, as I mentioned and alluded to before, we assessed along a number of assays, all available within the LINCS consortium. And those assays all give us readouts in signaling behavior, protein expression, transcriptional changes, epigenetic changes, and behavioral changes. And so this provides a really deep view into how cells are responding to these different ligands. And again, this was coordinated across the LINCS centers in great detail in terms of coordinating across batching, sharing reagents, sharing aliquots of cells that had been treated, and so on. So this was a highly controlled experiment that, again, could only have been done in the context of a consortium framework. I won't go into any more details on the experimental paradigm, but I think it's quite important and was very thoughtfully done. And there is a workshop later this afternoon that Sean Gross will spend some time getting into the details of exactly how and why we made the decisions that we did in the experimental data generation. So just to get into the data now and to give some views into what we have seen and what we are learning. is that the extracellular signals induce unique cellular phenotypes. What I'm showing you here are images of those MCF10A cells treated with each of the ligands indicated there and assessed after 48 hours. And what we can see is a striking change in the cell morphology by changing simply this single ligand in the media. We induce these very different molecular changes. We can then take these images and quantitatively analyze them in a variety of ways to confirm for ourselves what we see by eye. And for instance, we see changes in cell count, where EGF induces the highest changes in cell numbers, changes in the number of neighbors, that we see a striking change in this oncostatin M condition. The distance between neighbors we see varies across, and that with TGF beta sticking out as that condition which leads to cells becoming really flat, really spread out from neighboring cells as assessed by the center of their nuclei. And finally, we also see changes in keratin 5, which is a marker of differentiation. So the key observation here is that each of these single extracellular signals induces a variety of cellular phenotypes. It's not a one-to-one mapping, but of course, as we all can appreciate in biology, a many-to-many mapping, and that in and of itself provides a richness in the data and allows us to look across various dimensions and to analyze the data in a variety of ways to look for patterns and leverage that many-to-many mapping. Just getting into some of the molecular data and give a tiny view into that. What I'm showing you here are the RPPA proteomic data. One of our favorite representations, the heat map. Along each column is a single sample. Each row represents an individual protein as measured by an antibody readout. The data are organized to show, first organized by ligand, shown by the rainbow bar at the top, and then organized by time point from early to late. And what we see is that across the RPPA data set, we see a shift in the protein landscape as that shift is dictated by both ligand and time. We see some shared changes as well as unique changes to each ligand. The callouts there indicate some of the key canonical changes we see downstream from some of these ligands. And we see some of the canonical changes we expect, for instance, downstream from interferon gamma. We see changes in IRF1 and PDL1. Those are known downstream targets. And we see also downstream changes in phosphostat and so on. So there's some things we see in the data that give us assurance that the data are high quality and telling us some of the things we expect. And so then that tells us that when we start to make observations that are novel or surprising, that they carry some weight. The other thing to note in here is that we also performed for each of these ligand time points replicates across all of the assays, at least 3 replicates, in some cases more, depending upon exactly the needs of the particular assay. And by and large, what we find is that the replicate to replicate variation is quite modest. This was a very well-controlled experiment, so we see fairly modest replication variation. Worth knowing that these are true biological replicates. For those in the audience who are experts in understanding that biological replicate is a continuum. What we find is that responses to these extracellular stimuli are encoded at all molecular levels. What I'm showing you here now are data, an analysis of the RPPA data, where we performed a Maverick analysis, which is essentially a way of identifying what signal in the data can be attributed to the different experimental variables. So what we find in the RPPA data is that about a quarter of the variation in the data that I just described to you can be attributed to the ligand treatment. Another 10 to 15% can be attributed to the time point. So there is this dynamic evolution of the response. As I alluded to, the replicate variation is quite modest. There is signal in the data that we cannot explain by any of these single features. In many cases, this unexplained variation actually results from signal that can be explained by not a single ligand perhaps, but by multiple ligands. If we look across the data types, what we see is a similar result, which is that across each of these different assay types now, each of those bars being a different assay type, there is signal attributed to ligand or time point. It varies a little bit from assay to assay, but we see it in each of these assays that we ran. We do see the greatest signal in terms of ligand treatment in the RNA-seq and the ATAC-seq data. Worth noting that those are the two densest assays in terms of the numbers of readouts. They're the two most unbiased assays, and so we think that is a good portion of the reason why we see those giving us a greater signal. So what I want to do for the rest of my time is to tell you a little bit about oncostatin M and to use this as an illustrative use case. And one of the most striking things that we observed about oncostatin M from our live cell movies is that it induced this very striking phenotypic behavior. What you're looking at here on the left-hand panel are EGF treated, on the right-hand panel are oncostatin M treated MCF10A cells. So the exact same cell type, when we treat them with these two different ligands, we see a very different behavior. You see going across your screen in oncostatin M now, this large group of cells undergoing collective migration. This phenotype we thought was quite unusual. It's one that we saw only in the oncostatin M treated condition, and one that we've been following up now in terms of trying to understand what may be the molecular processes that control this phenotypic behavior, and this quite complex phenotypic behavior, I might add. And so in order to set down this road, what we did first was to identify those molecular changes that are induced by oncostatin M. And what I'm showing you in that bar graph there if we simply go through each of these assays feature by feature and ask, did oncostatin M strongly upregulate or downregulate this feature? Then you get a notch in the bar. And so what we see is that in the RNA-seq data, there's a very large signal associated with oncostatin M. We also see changes in these protein-based readouts in the RPPA and cyclic IF as well. Those bars, of course, are shorter than the RNA-seq. As I mentioned earlier, we don't have quite as many readouts in those assays, but nonetheless, there is a robust signal in those two assay types. And what we wanted to do was to get beyond an analysis of just looking at lists of features and move toward a more comprehensive integrative approach. Again, this concept of integration, which is something that has come up in the earlier talks, but to integrate across these different readouts. And so to do that, what we have done is a causal path network analysis, which is shown schematically down at the bottom here. What we do is take a set of curated pathways, marry that with the molecular data that we measure, and then use that to infer the network structure, which is shown there down on the lower panel. Once we do that, we can generate one of these very large hairball networks, which I'm showing you on the right-hand side. Each node represents the inferred expression of an individual protein. The lines between them are edges, which indicate relationships between the proteins. The color of the node indicates whether the protein is up or down regulated after oncostatin M treatment. What we have now set out to do is to start to analyze this network in a variety of ways to identify some of the key features, key molecular changes that are induced after oncostatin M treatment. And one of the most striking things that we see is this most highly connected node is STAT3. So as zooming into that network, what I'm showing you there is the location of STAT3. If we take that idea as a hypothesis that STAT3 is modulating oncostatin M phenotypic responses, we can go back into the lab and run an experiment where we take cells, and this is, again, that feedback arrow. We take cells and treat them with either oncostatin M or with oncostatin M plus a JAK-STAT inhibitor, and then ask, how well do cells grow over the next 2 days after we treat them with those two sets of agents. And what we see is that in oncostatin M treatment, cells are proliferating quite happily. You see that black line heading up. And when we add on that JAK inhibitor, we end up suppressing that proliferative response. So telling us that this association we identified with STAT3 is causally related to the oncostatin M phenotype. Surfing through the network a little bit more, what we find are strong signals related to cell-cell and cell matrix junctions. I've highlighted a few of those here. Cadherin, integrin beta-4, GJA1 is a quite intriguing hit. This is also known as connexin 43, thought to be involved in forming gap junctions, which are tight junctions between individual cells. The idea of cell-cell and cell matrix connections are quite important if one thinks about the requirements for collective cell migration. This requires that cells are both sticking to one another and also interacting with their cell surface so that they can be moving across the surface of the dish. Follow-up, more detailed imaging experiments shows us that there is some evidence of these oncostatin M treated cells forming very tight cell-cell connections and also interacting with the cell surface. What you can see in this particular image are these really bright spots. These are cells treated with oncostatin M and then stained with a marker to visualize actin, which is part of the cytoskeleton. And what we see is that these cells really are forming these very tight junctions. You also see these protrusions around the edge of this cell cluster of cells interacting with their with the cell or the surface substrate surface.
1 minute warning.
The last set of experiments I want to tell you a little bit about are related to the activation of various proteases. So within this network, we find a number of proteases. I've called them out here with the arrow highlights: MMP3, MMP14, HP, and a whole collection of serpine family members. These proteases we thought are quite intriguing hits. Again, if we think about this concept of collective migration, it turns out for cells to do that, what they need to do is to clear a migratory path. And in this particular case, cells are grown on a collagen-coated plate. And so what we hypothesize is that cells actually, in order to migrate around, need to sort of chew through this surface in order to make way for the cell to migrate through. And indeed, if we come in to go back into the lab and run an experiment where we take those oncostatin M cells or oncostatin M treated cells and add on a broad spectrum protease inhibitor, what we find is that we still see the cells form clusters, but these clusters no longer migrate. You see now in this dish that they're just sitting there. This is going to loop around a little bit, so you'll see this coming up again. So we still, the little jumping is just an artifact of the imaging, but what we see is that they do still form clusters, but these clusters just sort of sit there. So telling us again that implicates the protease activity as a key mechanism modulating this phenotypic behavior, this clustering migration induced by oncostatin M. Again, going through the network and thinking about what are the other processes that may control this behavior. What we find is strong signals related to CEBP transcription factor networks. In this case, the network, as I mentioned earlier, was built based on RNA-seq and protein data. We have additionally held out data from ATAC-seq that tells us about chromatin, which is intimately related to transcription factor activity. If we do an analysis of the ATAC-seq data and look for enriched motifs, what we find is that one of the top hits for oncostatin M is in fact a CEBP ATAC-seq motif. So this is a different way of thinking about validating what's in the network. In this case, the attack seek was essentially a held out aspect of the data that we can then use to ask, is this consistent with some of the other data that we see, and consistent with some of those other signals that we see coming up as hits? And indeed, that is the case. This particular factor we think is quite an intriguing hit to have come up upon, because it is known to be induced by STAT3. It's also been implicated in epithelial cell migration. And finally, its downstream target genes include many proteases, including some of those I showed you on the previous slide. So this, we think, gives us a way of trying to link across some of these different readouts to identify some of those key nodes responsible for driving this complex phenotypic behavior.
We are 2 minutes over time. We'll have to move to the next speaker.
Thank you. Just in time, I'm ready to wrap up and give you the summary and conclusions for this, this project. What we have found is that extracellular signals induce complex dynamic phenotypic responses that are encoded across all molecular levels. That perturbation biology is a powerful approach to identify molecular correlates of complex phenotypic responses. And that we believe these studies really set the stage for understanding responses to more complex stimuli, such as ligand combinations. And finally, our observations imply that responses to other perturbations of interest, for example, drugs or mutations, are also modulated by extracellular context. This is a topic that Jim Corcola will talk about in tomorrow's morning session. And we believe these rich, high-quality data will serve as a resource for others to build integrated multi-scale models of cellular networks. And finally, the data are available on Synapse with this ID shown there. And both Sean Gross and Mark Dane will provide later today a tutorial on experimental design and data access in one of the workshops. And with that, I would like to again just acknowledge all of those across the Links Consortium who contributed to this project. Certainly a tour de force, and I feel privileged to have had the opportunity to tell you about it today. And with that, I will hand off to the next speaker, and I'm happy to take any questions during the Q&A session.
All right, thank you very much, Laura. Our next speaker is Todd Golub, who's going to be presenting a talk on using perturbational data to inform mechanism of action and drug repurposing.
Thanks, Steve. Hopefully you can see that. It's a great pleasure to have the chance to update everyone on what our center's been doing, and it's been great to see how the links concept has been evolving and maturing over the years. I have a number of advisory roles in some companies that are listed here, but I won't be speaking about any of their products. So first, let me start with a little bit of philosophy around how we think about the links concept in what we've been calling the connectivity map. The idea that if one could represent disease states and perturbations, for example, genetic perturbations or drug perturbations in a common language, then one would be able to establish connections, thereby calling this a connectivity map, between biological states, cell states, and either pharmacologic or genetic perturbation states. To get a little more detail on how we think about this, I think actually that the links, that the L in links stands for library, and I think it's useful to think about this in a, what would you like to have of such a library? And those attributes are listed here. One is that it should be comprehensive. That is, addressing a breadth of biology, kind of like you think about a comprehensive Library of Congress. You'd like to have information readouts that aren't too bespoke to any particular biological system or question. You'd like it to be really easy to look things up in the library. So you'd like a card catalog of sorts. And as I think a number of other speakers have mentioned this morning, you'd like to have this library not be too hardwired to any particular experimental platform. That is, you'd like to be able to navigate between links data and non-links data. And you'd like this whole thing to be accessible both to computational savvy scientists and to biologists who aren't going to write their own code to analyze the data. So in a little more detail, of course, the concept of this links connectivity map, it has three basic components. The perturbagens, which again can be overexpression or loss of function genetic perturbations, small molecules, or in principle, large molecules, although we haven't done that much with biologicals, or could be small molecules that have become drugs. There are the cells themselves that are perturbed, and then some readout. And for most of the discussion today, I'm going to focus on gene expression as the readout. We, in the pre-links era, piloted this concept of a gene expression profiling as a readout of cellular state with 154 drugs profiled in a few different cancer cell lines, which was fine. But the links program has allowed us to expand this now to the final links data set, which contains about 3.1 million gene expression profiles that are all now publicly available. And I'll expand on this a bit more. In order to make such a large scale library feasible, the approach that we took was to say that if the business that we're in for this application is to match the gene expression signature of one particular cell state to another cell state as induced by perturbations, then perhaps we don't need to measure the entirety of the transcriptome, and perhaps we could measure a subset of the transcriptome, which would be sufficient for inference of the entirety of the cell state, or perhaps even the entirety of the gene expression profile. And without going into detail, we selected 1,000 transcripts out of the roughly 20,000 protein coding transcripts that we call landmarks, and found that that would recover, based on analysis of our pilot data, where we had the genome-wide expression data, about 80% of the information as assessed by this connecting one cell state to another. We then developed a method which we called L1000. This is the work of David Peck in the laboratory, whereby working from crude lysates of cells grown in 3D4-well plates, we can do locus-specific 1,000-plex amplification of these landmarks, and then detect them by hybridization to fluorescent microspheres or luminex beads, thereby getting simultaneous readout of 1,000 expression measurements per well of a 384-well plate. This slide simply makes the point that, of course, as you'd expect, there's a lot of data processing involved in getting from raw data all the way to a computational assessment of a connection between one cell state and another. Because there are so many steps to this, we made a deliberate effort to make data at all of these levels publicly available, recognizing that, for example, the most computational sophisticated individuals in the community might have ideas around how best to process these data, the raw data, whereas mere mortal biologists would like to have much more highly processed data. And so making data available in a transparent way has been key to this effort. I'll also say, and there will be a poster later today on this topic, that we've also had a number of crowd challenges to engage the international computational community to address not the entirety of this links challenge, but focused efforts. For example, how can you best go from 1,000 measurements to inference of the entire transcriptome? And these have been interesting and productive challenges. We've now published the first million or so of these links profiles on the L1000 platform, as shown here and described in the paper in Cell a couple of years ago. I'm pleased to say now that we have the final links data set in hand and out of hand, that is in the publicly available, now over 3 million profiles that represent about 92,000 different types of perturbations. For example, more than 25,000 small molecules, including many drugs and many non-drug small molecule tool compounds. And those have been explored across a large number of different cellular contexts, primarily cancer cell lines, because those are the most tractable and widely available across lineages, but also including a number of primary cell types. Of course, all of those 92,000 perturbations have not been conducted across all 253 cell types. So it's not a completely filled in matrix, but it's a very rich data set. Now, one of the interesting ways to use this integrated genetic and pharmacologic perturbational data set is to ask questions such as, to what extent are genetic perturbation and pharmacologic perturbation biologically equivalent? That is, do they induce the same gene expression signature? And in many cases, as shown in the top, and the representation here of this triangle represents a compound or CP induced signature, a CRISPR knockout signature of its intended target of that compound, and CGS for a consensus gene score reflecting a summary score of multiple shRNAs targeting the same gene, again, the target of the compound. And you can see for the examples shown at the top, we see a strong similarity in gene expression signature across these three different types of perturbational measurement. In this case, suggesting that the consequence of perturbation with a small molecule compound, for example, an mTOR inhibitor, is very similar to genetic perturbation of mTOR as a specific target. In other cases, we don't see this, what one might think would be expected strong connectivity. We might see connectivity only between a compound and complete loss of function with CRISPR, but not with what's typically partial loss of function with an RNAi. Sometimes we see strong connections between the two types of genetic perturbation that is not recapitulated by the compound. And I think it's difficult to, in summary form, explain this entirely. I think it's fair to say that we now believe that this is a combination of technical features, for example, the selectivity of the genetic perturbagens and their off-target effects, the selectivity of the compounds and the dose at which they're tested, and also the fact that in many cases, complete loss of scaffolding function of a target biologically is not the same as enzymatic inhibition of a particular function with a small molecule. And so the links data set is recapitulating that difference between those two different functions. We've also learned, although I don't have time to get into it in detail here, that while we tend to think of small molecules as inhibitors of their particular target, there's now abundant evidence that even highly refined and optimized small molecule inhibitors also have additional unexpected function, for example, functioning as molecular glues, where they're stabilizing protein-protein interactions that themselves will have downstream gene expression consequences. Now, what about using links as a way to discover new mechanisms of action of small molecules? And I'll show one example here, focusing on the target casein kinase 1 alpha, CK1 alpha, which is emerging as an interesting target both in heme malignancies, in part because it was shown by Ben Ebert and colleagues to be the target of lenalidomide, and it's clinically efficacious in 5q minus myelodysplastic syndrome. But there aren't great CK1 alpha inhibitors available. So we said, well, could we just look one up in the links data set? and ask, if we make a signature of CK1 alpha loss of function genetically, in this case with RNAi, what is the top scoring small molecule that matches that CK1 alpha genetic loss of function signature? The compound is shown here. When this was then profiled with standard kinome profiling approaches, as shown here, we're able to see that this compound, previously unannotated and not optimized for anything, was actually a quite selective inhibitor of casein kinase family members. And moreover, we could show biochemically that the compound was indeed a CK1 alpha binder and inhibitor as predicted by the links analysis. This then made it possible to run with this hypothesis that this compound discovered by links was a CK1 alpha inhibitor. It was co-crystallized at high resolution, as shown here, providing structural evidence for how it is that this links-derived compound was in fact functioning as a CK1 alpha inhibitor, and also providing guidance for further optimization. And we've now partnered this CK1 alpha program with a major pharmaceutical company to drive this toward optimization for the clinic in the future. Now, I've been focusing on L1000 gene expression as a type of perturbational readout. You're going to hear shortly about proteomics efforts, and we've heard also a bit about using imaging as a readout. I want to draw everyone's attention to one additional type of perturbational readout, and that is that of cell viability using a method developed by Channing Yu, a postdoc a former postdoc in the lab, that we call PRISM. And PRISM, the goal of PRISM is to say what we'd really like to look for is subtle perturbational signatures or profiles across a large number of cancer cell lines, thereby allowing us to look for correlates, genomic correlates of that subtle behavior. And we do this rather than measuring viability one cell line at a time by barcoding them, by inserting randomly in the genome a random sequence, 24 nucleotide barcode sequence into the genome, pool the cell lines together, and then can subject those pools to perturbation, either genetically or in this case with small molecule drugs, and then use the barcode abundance, which we read out either on Luminex beads, similar to L1000, or by sequencing, and then use that barcode abundance as a proxy for viability. We've now barcoded close to 1,000 human cancer cell lines, and I'll show you the recently published results of the first analysis of that large PRISM data set. This is the work of Stephen Corsello, a postdoc in the lab, where Stephen profiled about 4,500 drugs across 578 cell lines. These are not just cancer drugs, actually mostly not cancer drugs. And then selected for follow-up those compounds that showed some viability effects at a high concentration, followed up with dose response across the PRISM panel. So what's shown here is a UMAP representation of the similarity of these compounds to each other, not in the space of structural similarity, but rather in the space of similarity across this panel of 578 cell lines, their viability effects across 578 cell lines. And I think what's particularly interesting here isn't so much that you can efficiently recapitulate the effect of known oncology drugs, which you can, for example, the tubulin stabilizers and destabilizers, but that you also can see the similarity and perturbational response across non-oncology drugs that have very subtle growth inhibitory activity, for example, glucocorticoid receptor agonists, not suggesting that those are interesting oncology drugs, but using these subtle differences in viability changes to show that they must have a similar mechanism of action. And in experiments I don't have time to go into, we're able to use this approach also to discover new mechanisms, for example, a new molecular glue that results in degradation of cyclin K, work done in collaboration with Nico Toma and Ben Ebert, also recently published. Another example showing that these small molecules, in this case, a small molecule called CR8, developed as a kinase inhibitor, is actually functioning as a molecular glue, resulting in degradation of a downstream target, cyclin K. So I think the true measure of the utility of these kinds of data sets is whether other people are using them. All the data that I've described here are available on CLUE, which is the data repository and compute infrastructure we've implemented on the cloud, but the data are also available from GEO, and analysis also available from the LINCS DCIC Analysis Center that you'll be hearing about shortly. The statistics shown here simply show that people are using the data, as Ajay showed in his presentation. There are a number of users of the data and the tools and combination. What's gratifying is that we do, as we'd hoped, see a blend of computational and non-computational users. Perhaps too many people still downloading the data, which I think means that the tools that we've developed aren't yet sufficient for all the analytical interests of people, because in the fullness of time, I think we want to make people bring their tools, or have people bring their tools to the data rather than downloading these growing data sets, but it does at least look like people are using the data. In the last 1 minute, let me just say that one of the concepts of having a large data set is that one might be able to turn to it to rapidly ask questions that are pressing. We've started to do this in the case of COVID-19, recognizing, for example, that the receptor for the SARS-CoV-2 virus, ACE2, might be a target for therapeutic intervention. And so we asked among the LINCS cell lines that had been profiled.
1 minute warning.
What's a cell line that has expresses reasonable levels of ACE2? That is BICR6. We then looked in the BICR6 LINCS data, which had been profiled across about 15,000 drugs and some tool compounds, and are now testing those compounds that, based on the LINCS data, either downregulate ACE2 or the downstream program of LINCS2, and we're testing that in first a pseudotyped lentiviral assay for SARS-CoV-2 infection, and then anything that survives that assay will be tested in a live, real SARS-CoV-2 infection assay as well. So too early to tell whether we'll get something out of this, but it's an example of rapidly using data computationally to generate new hypotheses. Let me end by acknowledging the people that have really led this work, in particular, Arvind Subramanian, co-PI on this project, Rajiv Narayan and Ted Natoli, who will be giving poster presentations and workshops, Stephen Corsello and Jennifer Roth on PRISM, and a great new team working on COVID-19. Thank you very much.
Okay, thank you very much, Todd. We're on to our next talk. We hear from Clive Svensson, establishing an iPS-based multi-omics and cell-based pipeline for motor neuron disease.
All right, can you see my screen okay, Stu?
It looks great.
Yeah.
Cool, you can see yourself there. So first of all, it's been great to hear the updates from everybody this morning. We're going to switch gears a little bit and get to the NeuroLinks Consortium, who are shown here on this slide. And this has been an amazing collaboration across the LINCS community, but also between the institutions mentioned here, Leslie Thompson, Jeff Rothstein, and Steve Finkbeiner, along with Jenny Drew and myself at Cedars, and then Ernest Frankel at the bottom there, who is now joining us, as you'll see in the second part of this talk, as part of Answer ALS, and has been really instrumental in pulling all the data together. And it's going to be sort of two parts. Today I'm going to be talking more about the iPS modeling, and then tomorrow Leslie is going to take the baton over and continue on with more of a deep dive into the multi-omics approaches that we've used to analyze this data. So if I go to the next slide. Disease that we're talking about is the one you really don't want to be diagnosed with. This is a pretty horrific disease, Lou Gehrig's motor neuron disease, and the early signs is twitching and fasciculations. Then you get death of the motor neurons, both up here in the upper motor cortex, that then connect down to the lower part of the spinal cord to have the second circuit, which goes to the muscle to produce movement. Really are no drugs. Riluzole and edaravone only extend life by about 3 or 4 months, no cure. As for many neurological diseases, 10% are familial. We know the genetics of 10% of ALS, but about 90% we don't. I'm going to come back to this in a second with that C9orf mutation. And so it's very difficult to develop drugs when you don't know the target. We do know a lot about the mechanisms of degeneration using animal models, basically by overexpressing some of those mutations I discussed. And that's led to a really complex view of ALS. Many pathways are disrupted when you perturb different parts of the system of the motor neurons, either the upper or the lower. But in essence, we don't really know which one of these are causative, which one of these are the original problems that cause the disease in humans. So we desperately need to know more about these issues in human cells and not animal models, and then integrate all of these changes some way to understand more about the disease. As for sporadic ALS, these forms that don't have a genetic, we really don't know much about the cause at all. And so for those, we need new models. And this really brings up, we heard from earlier on about the heart iPS-derived cardiomyocytes. Unless you've been under a rock for the last 15 years, you all know about iPS cells. And this is where we take from a diseased patient the cells, reprogram them. We use PBMCs, blood draw, use the Yamanaka factors and get them to reprogram where you can make any cell of the human body. And as you heard before, Ravi is using heart cells. We use the same technology to produce brain cells, neural cells from these tissues. I think traditionally we're thinking of using these cells for transplantation, perhaps, but more recently I think the legacy of iPS cells is going to be disease modeling, because you can create the lines that represent the patient's genetics. And so through that, we can learn about a lot more about the origins of disease that have a genetic base. In other words, if there's a perturbagen that causes disease, we should be able to discover it in these iPS models. Using iPS to create motor neurons, we've reviewed this literature quite extensively recently. Essentially, normally when you make motor neurons, this is the in vivo part here. You go through gastrulation, neurulation, all the way through to the spinal cord where the motor neurons exist. And I'll focus today mainly on the spinal cord motor neurons. And we've mimicked that in the vitro, in the Petri dish, where we essentially take iPS cells, take them through the same initial parts of gastrulation and neuralization by using Wnt inhibitors, dual SMAD inhibition. And then with retinoic acid, sonic hedgehog, we go down here to get something that looks very much like the spinal cord. So on the top is a human spinal cord, and on the bottom are our motor neurons generated from iPS cells. So through a lot of work and a lot of community effort, we have a good way of making these cells. Now, when I came into this field, 2009, and very first iPS modeling papers were coming out, we decided to look at this in a very severe motor neuron disease called spinal muscular atrophy, which occurs when, in babies, it's a horrific disease. They go floppy after about 2 years or 1 year and become completely paralyzed. And we decided to model this by taking iPS from SMA patients. We made iPS, and just as I've shown you, we differentiated them. And in SMA, we actually found that the motor neurons after 6 to 8 weeks actually began to die in the Petri dish, just like they did in the child. And so this is the first evidence that this modeling can be incredibly powerful. That led to actually a lot of drug screening, and more recently a new drug from Roche that's now being used and approved for SMA. But the real focus for us was on adult diseases, and the most common adult disease causing ALS is caused by repeats in the exonucleotide repeat, so the so-called C9orf72 gene. And if you have repeats in this gene, it can be up to over 1,000, you get the symptoms of ALS, Lou Gehrig's disease. And so we made iPS cells sometime, Jeff Rosting's group and our group, and now many others, and showed that you could generate motor neurons from these C9 patients. And unlike the SMA, we didn't see overt neuronal death in these models. We knew there must be something in the genetics was changed, but we didn't see the cells die. We did see a number of DEGs, differentially expressed genes, in these papers. And what was interesting is because this is an early motor neuron, it does represent the very earliest stage of C9orf-induced ALS. And so we could pick up these signals. So the idea for Neuralinks was to look at these signals at a wider level to try and understand more about these initial molecular changes using this model, and that way become, use more of an understanding and design new drugs. And this is the consortium that we funded. We were in the second phase as well, along with Sinai, kind of Sinai and Mount Sinai and Cedar Sinai, and the collaborative groups involved. And so we've been very fortunate to have the Links funding to push this work forward over the last few years. And this is the website, and ultimately you can just go look at neuralinks.org and get caught up as we come to the end of this project. We've downloaded a lot of information here, and essentially the idea is to take motor neurons that I just showed you and then apply transcriptomics, proteomics, epigenomics, and imaging, robotic imaging, to the same batch of cells. So to get a large view, high view, using multiomics on what goes on in these motor neurons, more or less to get at that problem that I showed you at the beginning of what are the underlying changes that happen in these motor neurons in order to understand more about how that triggers the cell death downstream. And then use network analysis and ultimately perturbations, which is just starting now on the cells to understand more about the disease. For the original Neuralinks program, we used 4 C9orf ALS cases of 4 controls because of cost and the amazing complexity for differentiating these cells, as you heard earlier, for the heart. This isn't a typical Links perturbagen study that we were watching from the other groups. You just heard from Todd, where you screen thousands of molecules, small molecules. Essentially, this C9orf is genetic perturbagen in these patients. And we went in from the other side and said, could we understand with this one genetic perturbagen in this iPS model, what the downstream cascades are that cause the disease, and thus understand more about how the disease works. Tomorrow, Leslie is going to go through some of the findings from the 3 or 4 years work. It was an enormous amount of collaboration and work between the groups I mentioned. We did find that there were causal pathways with the C9orf mutation, but interestingly, people don't consider so much, there were compensatory pathways. When a perturbagen hits a cell, it doesn't always kill it, even though there's a pathogenic gene involved. In some cases, what you measure as an output is the response of the cell to try and protect itself. And through the multiomics approach, we were able to untangle both compensatory pathways and causal pathways that were leading to the downstream changes with this one C9orf mutation. And we posted this on bioRx. It's under review at a journal currently. We really hope to have this out in the general public very soon. So we conclude from this that this multi-omic approach, we could look at the specific cascades that lead to new therapeutic targets. There'll be more details from Leslie tomorrow in her talk. But I'm going to transition. The next question really we had was sporadic ALS, which really represents most of the cases. And the answer to the question here was, could we use the same kind of multi-omics approach that we built through Neuralinks to see if a genetic perturbagen is seen across all of ALS and not just where we know there's a mutation. And this really led to Answer ALS. Now this rides on the shoulders of Neuralinks. And for the last 2 years, we've really, and 3 years, interwoven the 2 projects. But it really shows you how this NIH funding from the Common Fund has allowed us to leverage, and I made the NIH signal emblem a bit bigger here. It was really this confidence that we gained from the Neuralinks community and the Links community in general that allowed us to convince a number of donors to scale up. Because 4 and 4 lines is just not enough. And yet when we get to more, it's very expensive. So we had the original Links team here, and then we combined with a lot of clinical teams. And I think what we found in Links, somewhat frustrating, was it's nice to have an ALS line, but you really need to know more about that patient and the underlying biology of the patient in order to understand more about the disease. And so we embarked on a very ambitious project to analyze 1,000 patients. So we through a number of clinics listed here, we did generate samples. We collected samples from 1,000 patients. through the New York Genome Center, we did whole genome sequencing. Here at Sinai, we made iPS lines, I'll talk about in a minute, during that I've shown you before, and I also differentiated them into motor neurons. And then we collected a lot of clinical data as well from these patients. And the goal here is, in these 1,000 patients, could we see genetic perturbations within those cells that created subtypes of ALS? And so the hope is that if a bunch of patients went in different, luckily here they're different colors, the assumption is there'll be subtypes of ALS. When we get this signature at the end, we can actually combine this information through multiomics and advanced data analytics with all the signals that we get, combine it to produce subtypes. And if we have subtypes, we can develop drugs against those subtypes. And of course, those drugs then, Precision Health, will go to the appropriate cluster of patients that we collected originally. Now, once we've done this once and we've got the database, we hope to enter patients then into a new trial where very quickly we'll assess what phenotype they are and put them on the right drug before they progress with their ALS. And obviously that's the goal. How do we do this? And I think this is relevant for this family of links folks on the call. It is hard to make 1,000 lines, and we had to standardize a lot of stuff. And I'll mention briefly in the last 5 minutes how we kind of have gone about that. First, we collect the blood sample from the clinics, and this was 5 clinics across the country, shipped to Cedars. The PBMCs were reprogrammed, which takes around 90 days. And then we made the iPS lines, which we stocked. We do make them in a way that can be distributed to others later on. So they're saved and stored, but we also then differentiate one set in one protocol and then do the data analysis. Just to update from Answer ALS, we're currently here somewhere, and we've made about 658 iPS lines as of this week. We got a little delay with COVID, but we're back on track. Just to point out, Lindsay, Panther, Lauren, O'Neilis, and Drew Serene and their teams at the Biomanufacturing Center here at Cedars have been critical to make this happen. And again, Drew and his team and myself and our team developed this protocol with a lot of help from the community, where it's essentially a 3-stage protocol to 32 days to go from iPS cells all the way through to motor neurons using kind of standard tissue culture techniques. And then for links, what was interesting is how do we get these samples out to all of the sites for Answer ALS and links based on the links program. And here we just took 6-well plates and we actually distributed exactly the same sample into 3 different aliquots. And one went to mass spec with Jenny Van Eyck, the other went for RNA-seq with Leslie Thompson, and the other went TAC-seq at MIT through Ernest Frankel's group. And so these 3 samples were all identical from an identical tube, which means we can cross-compare all of the omics information across those samples. And the other challenge we had was consistency. So we decided to do, we ran these in 12 batches of 12 samples. Sorry, I'm going to go back a second. We ran these in batches of 12 samples, 12 patients rather in each batch. So we ran them all along together for 32 days. And each time you put one control, which means the same line was run each time. And then we had a technical control, which was one huge number of, huge batch of motor neurons that was made in order to run at the same time. So that represents the same exact tissue being run on the omics analysis, whereas the batch control is one line that was run along with all the others through the differentiation protocol. And we've already done this now with 350 lines have already been distributed and differentiated. This is Drew's data where he differentiates, and we use molecular dynamics machine to quantify the number of neurons that come out. And you can see here the kind of images we get for markers for motor neurons. You can see all the axons here spreading between the clusters. And nestin is a progenitor marker, and then these other ones are neuronal markers. We basically take all of these markers and analyze them across, at the moment, this is 172 ALS lines and 32 control lines. And the plots are shown here. And in fact, just the raw numbers, the differentiation are fairly similar between ALS and control. So you can see there's no significant difference except for a slight increase in ALS versus control with islet-1. And this is actually a marker for motor neurons, which was interesting to us that we'd have an increase. And this sort of suggests or hints at an overproduction and an overproduction perhaps of these early motor neurons early, and maybe there's a severe loss later. And so this is an exploration. We're going through this process of exploring the mechanisms under that now. Other markers were not changed between ALS and control. And then the very, very preliminary, and this is work that postdoc in my lab has been working very closely with Jenny Wu, Ryan Lim, and Leslie Thompson, who generated the RNA-seq data. Just simply, can RNA-seq separate controls from ALS, doing just a non-biased PCA? And we found no, controls in ALS are pretty well mixed with just a basic PCA analysis of the RNA-seq. However, you can clearly see the two groups here have emerged, the top one and the bottom one. And when we just looked at this in simple terms and just did some covariance, it's clear that males on the top and the females are on the bottom. And so something that's come up and come to our attention, and I think Leslie will talk more tomorrow, is that male-females are very important in these analyses. And I think with the heart, you heard that you have to have a mix from male and female. And we were surprised that neuronal cultures vary so much solidity between male and female. And so we had to rethink comparisons between male and female and separate them out. And if you do that, the noise becomes a lot less. We'll hear more about that tomorrow. And actually, when you break down the counts now, you find that the male counts shown here for ALS were slightly higher than the female for ALS. And so this represents some of the difference between male and female for ALS. Males have more islet-1, more motor neurons, maybe because they need more. We don't understand that yet, but this is just coming out of non-biased counting of all the data. The batch technical controls, I mentioned, we did make a batch of technical controls. One line that we just made a whole, like 150 tubes from, that we ran with every sample. And then we did a batch control that was run with each sample, what we call a differentiation control, BDC. In other words, the same line was run with every batch that we ran. And you know, the question is, how consistent are the numbers? And these are just the batch controls plotted over time. And you can see there's variations in nestin, which is a progenitor marker. And so that isn't held very consistent. But TUJ1, which is a neuronal marker, every time, these are now up to 38 batches with 12 lines in each batch. The neuronal markers are holding very, very tight across batches. And so the same line is giving the same results each time, so that we can anchor now our data a little bit in the batches. The technical control at the RNA-seq level was like a bullet. So if you just take the same tube and keep doing RNA-seq, this represents variation in the RNA-seq technique, which is very small. And these purple dots are now the batch controls differentiated 25 times. And you can see there are differences in the PCA analysis over the different batches. We did a heat map of this, and here are the batch controls matched with each other. And you see they correlate very well. There's still good correlation of the other batch controls, but not as good as the technical controls. And this is the variation here. And the question is really which genes are affected by the variation. I'll just finish this piece off, and because I want to get on time, I know we're a little late. But essentially the bottom line is, if you do compare the mean expression with the variance, you expect it to get lower as you go up in the expression levels. And it does, and it correlates nicely in the technical controls. The differentiation controls, there are a number of genes which are disrupting this curve and pulling things out. So we asked the question, what things are variant across these batch differentiation controls? And it turns out to be mainly extracellular matrix genes which are driving that variation. So we think it's, we might be able to use that information to control for that and actually correct for that across all these lines. If you want to get these lines, thank you. If you want to get these lines, you can go to see the Sinai .org for biomanufacturing, and that way you can just get the lines that are in the repository. And you can also go online to answerals.org and look at the data that we're generating here. It's a fantastic website put together by Ernest Frankel and his team that is available right now to go look at. And you can spin through these 1,000. We haven't got 1,000 up yet, but as they come, we're updating this website, and we're actually putting a newer portal together now as well. So the future, I think Richie Ho in my lab, who now has his own lab here, has been working hard. We do have to dissect out more within these complex cultures what's going on. And with single cell RNA-seq techniques now, we can actually focus in on the motor neuron group, which is shown here when we do single cell analysis, and just do our analysis on the motor neurons rather than the bulk cultures. And that's really showing us that if you just select, in this case, this is more recent work just using the frozen vase. Cluster 5 here represents the motor neurons. And you can see that the motor neurons, we can then just analyze separately, and we get much more resolution and genes going up and genes going down when we just look at motor neurons in these cultures. So I think that's kind of part of the future. And then the other part is newer techniques for differentiating the cells, chips, and we have transplants into animals, etc., to understand more and get them more mature. And I think this will lead to kind of this new era of medicine, where we recently were on the cover of National Geographic for the organ on chip models, but it just tells you that this field is moving very rapidly. And I think in the future, we'll be using much more sophisticated ways of differentiating the cells, combined with the links data to understand more about human disease. So I'll stop there. Very much acknowledge many folks involved, primarily from the CEDAR side. It was Drew Sareed and his team making the IPS lines, differentiating them, and then all the collaborators to do the omics. It's just the folks that really generated these 1,000 IPS lines and differentiated them. And then finally, the Answer ALS group that I mentioned earlier, and all of our links collaborators. And I'll stop there. Thank you very much.
Great, thank you, Clive. So let's go on to our next talk by Maeve Bonner from MIT, Unbiased Epigenetic and Phosphoproteomic Profiling of ApoE4 lines suggests novel biology and new potential therapeutics.
Can everybody see my screen? And it's in full presenter mode?
Yep.
Take that as yes. Thank you. Okay. Hi, my name is Maeve. I'm a postdoc in Lee White Tsai's lab at MIT, and I'm here representing a really fruitful collaborative project that has been between our lab with our IPS models that we have developed and the Broad Proteomics group with unbiased proteomic profiling, including GCP and P100. So I'm excited to tell you at a very high level the kind of the aims of what our collaboration is seeking to do, and also some of the very exciting ways we've been able to integrate these large databases to find out new things about Alzheimer's disease risk. So I will pointer. Okay, so I won't belabor this point too much, except to say that Alzheimer's disease is a devastating neuropathological disorder of increasing healthcare costs and increasing incidence with an aging population. And we have not been able to get to market new effective clinical treatments in over 2 decades. And this represents a real bottleneck in the understanding of not only what can we observe and learn about Alzheimer's disease, but how can we actually modify the outcome. So what we know about Alzheimer's disease, and Clive mentioned this in his talk, introduced it very well, so I won't have to talk about it too much, but there is a genetic underpinning to aspects of Alzheimer's disease, but the vast majority of Alzheimer's disease is considered quote unquote sporadic or late onset, which means that we have shown in this plot here genetic associations of risk from large population studies of Alzheimer's disease patients versus control age patients. And we see that variations in the genes that are shown here are associated with differential risk for Alzheimer's disease. The most highly validated, strongly enriched one is ApoE4. So ApoE4 is associated with a dose-dependent increase in Alzheimer's disease, where one copy of one copy of ApoE4 increases your risk for late onset Alzheimer's disease by about 3-fold, and 2 copies increase it by about 12-fold. So we clearly know that that genetic changes, mutations or polymorphisms in the genes in these plots are changing our risk for Alzheimer's disease. But what does risk mean at the cellular level? How can we understand why we have an increased risk associated with a gene like ApoE and the allele ApoE4? What are we saying at a cellular level about that risk? So what we were interested in trying to figure out is, can we start to say something fundamental about the nature of risk by doing combining our Alzheimer's disease models, so information from these genome-wide association studies that are identifying changes in the genome that are associated with changes in Alzheimer's risk or other disease risk, but not, there isn't yet a clear connection between those changes and the cellular outcomes of those changes. And clearly this is what this group is trying to do on multiple platforms. So we were asking as well, in our collaboration in particular, can we leverage these proteomic platforms to get insight into the processes and pathways that are either subtly or significantly changed in an Alzheimer's disease risk state, and maybe even potentially identify new means of intervention. So the way we do this is by taking advantage of iPS models. Again, Clive introduced these, so thank you for that. What we do here is we can take cells from either an Alzheimer's disease risk carrying patient or a control patient and reprogram them into iPS or induced pluripotent stem cells, and then we can generate these multiple different kinds of neural stem cells, so neurons or glia, as well as generating two-dimensional and three-dimensional models to try and understand the phenotypes associated with those. And a lot of these have been hypothesis-based to this point, because based on what we know about Alzheimer's disease, based on what we know from human brains and the pathologies we see, but maybe we need to take a more unbiased approach, or rather in parallel with these hypothesis-based approach, perhaps we can take another approach, an orthogonal approach, to understand in an unbiased way how just the cellular landscape is changing with respect to the presence or absence of these risk variants. And then if we can identify some sort of cell landscape changes and a signature associated with them, can we then screen against those signatures to identify previously unexamined drug targets? Oh, so actually, I also want to just plug here Priyanka Narayan, who is one of the valuable collaborators on this, will be talking in session 6 about the use of iPS cells in this kind of work. So the two ways that the Broad Proteomic group has been working with us to approach this question is by using global chromatin profiling, GCP, to do epigenetic profiling, or P100 reduced representation phosphoproteomic profiling. So the global, the GCP profiling, and I know everyone in this audience definitely doesn't need a much introduction here, but basically what we're looking at is histone modification, bulk histone modification, in order to try and identify trends of changes for potential transcriptional changes or regulational changes, and in phosphoproteomic profiling, identifying key sentinel proteins that could be representative of key pathway changes that are occurring in the cell types that we're interested in looking at. So the particular cell types we will be interested in looking at are APOE4 in cells that we will use to examine the difference that exists in an APOE4 risk state. So as I mentioned before, APOE4 is the highest validated risk factor for Alzheimer's disease. So what we did was we obtained cells from an APOE4 positive patient and CRISPR edited them back to be APOE3. So we have genetically identical lines other than at this APOE locus. And we did the same for a patient that was from a control population and was APOE3, CRISPR edited it to be APOE4. So we have two independent isogenic lines from which we can generate all of these different kind of neural stem cells. And we're using them for a lot of different purposes in the Scilab, but for the purposes of today's talk, and to take the greatest advantage of the GCP and P100 technologies, we focused on these two cells that I'm highlighting here, neuronal precursor cells, MPCs, and astrocytes. And we did that for a number of reasons, one of which is that these are both proliferative cell types, so they are amenable to these high throughput technologies, and we can apply them at scale to a lot of these questions. Another reason to look into these is that we've identified, and others have identified, key phenotypes in cells like astrocytes for APOE4, and this will give us an avenue to then do phenotypic exploration once we've mapped some of this landscape. So this is just the pipeline that we have put these cells through. So we've taken both NPCs and astrocytes from the 2 parental lines. So this is 4 lines in total, 2 APOE3, 2 APOE4, and put them through both GCP and P100 profiling. And then from there, and much data analysis and careful visualization of the data, tried to get to a point where we could ask, can we identify something as clean as an APOE4 risk signature with either of these technologies? So can we see something that is different about APOE4 between both of these parental cell lines? And then is this something that we can then use as a platform to understand more about APOE4 biology and risk, but also then potentially to push into new avenues for therapeutic exploration? So for the purpose of this talk, I'm only going to focus on, I'm just going to give a kind of case study. So I'll use the NPCs for the chromatin profiling that we've done, and astrocytes for the phosphorylation profiling that we've done. But at the end, I'll show a little bit about the discuss a little bit about the breadth of the work that the Linksborough group has been doing to generate huge amounts of profiles. What we're looking for here is we know some phenotypes associated with APOE4. We know a lot from human brains, but we're trying to figure out as well, not only can we identify pathways or changes that could help us to make sense of phenotypes we've observed, we also want to let the data tell us things that we are not expecting, and potentially new pathways to look at to understand APOE4 risk. So to begin with the chromatin profiling, the global chromatin profiling, what I'm showing you here on the left is the samples. Each column is a sample for NPCs of APOE4, APOE3, and you can see in purple and pink are the cell lines, so the originating cell lines. So the 2 purple are isogenic, and the 2 pink are isogenic. But what you can see is that the APOE4 cells cluster together and the APOE3 cells cluster together. So what we're identifying here are changes that are specific to the presence of APOE4 and not necessarily something that is occurring because of individual genetic background outside of this locus. Using this, and you can see there's pretty good reproducibility across the different samples. So then we did some careful marker selection to look at the marks that are changing most consistently across all samples and across both cell lines, as well as with greatest degree of change. And from there, we also normalized to the parental line. So basically what we wanted to see was just the difference that basically what was changing from APOE4 relative to APOE3. So what you can see here is the 2 APOE4 parental lines and the selected filtered marker selection, and this in the end this sort of signature of what is different in global chromatin profiling in NPCs in APOE4 compared to APOE3. And we are saying that this is actually giving us a really interesting insight into APOE4, and that it has a risk signature even at the epigenetic level. And this is something that has not actually been explored too much in an APOE4 research space, the epigenetic changes associated with it. So we think that this could actually be a super interesting new avenue for understanding APOE4 biology. So what we wanted to first look at was just, is there anything that this signature is just jumping out and telling us? And the overall trend seems to be increased acetylation and reduced methylation. And we're exploring sort of what that might mean now, but there is some literature evidence that supports that this could be a relevant outcome for APOE4 across multiple cell types, or rather in Alzheimer's disease, and perhaps APOE4 is contributing to this outcome. So to move to the phosphorylation profiling in astrocytes, we did essentially the same thing, which is we sort of followed the same data analysis workflow and looked at ApoE3 and 4 astrocytes by P100 profiling. And while the sort of just looking at the sample level, the immediate difference isn't quite as clear in astrocytes as in NPCs at the phosphorylation level, when we filter for the marks that are changing the most and most consistently across cell types, you can actually see that there's a very distinct ApoE4 signature compared to the ApoE3, and that if we now sort of normalize to each parental ApoE3, now we can see that there is, again, what appears to be a pretty distinct ApoE4 risk signature for the phosphorylation, P100 phosphorylation profile in astrocytes. So again, just taking a quick look at this, one of the things we're just trying to dig into, is this telling us something about new biology? So this THRAP3 is a thyroid hormone receptor associated protein, and there's sort of emerging evidence that thyroid hormone signaling could be pretty important in Alzheimer's disease and pretty important in neural and cognitive health in general. So it might be interesting if ApoE4 is significantly modifying this. One other P100 profile that came out is this reduced phosphorylation of this lima protein. And I highlight this just because some of the work that has been done on ApoE has been in its role as a lipoprotein carrier. So ApoE is known to modify plasma LDL levels, and this increases risk for cholesterol accumulation and other outcomes. And it appears that we're also here identifying sort of orthogonally differences in lipid metabolism associated with ApoE4. So we've successfully gotten to the point of being able to use our IPS models in multiple different cell types, multiple different assays to generate risk signatures that are starting to tell us something about the biology of ApoE4. But now we want to ask, can we use these the high throughput capacity of these screens to be able to identify novel means of intervention based on these signatures, regardless of sort of the biology that it's telling us? Can we just explore a drug space and understand whether we can modify it? So this is where the huge power of the links data sets and the skill of the Broad Proteomics group has really come to shine. So they've profiled dozens of cell types and across hundreds of drug profiles, and generated both GCP and P100 profiles associated with these perturbations with these drugs. So in particular, what we were interested in is this H9 IPS, basically an IPS sort of control cell, where we generated NPCs, astrocytes, and parasites and other cells for them. And then they profiled across GCP and P100 across hundreds of drugs. So we asked, could we compare our ApoE4 risk profiles that we received from the GCP or P100 to the profiles of perturbation in this cell type or in this cell line, and can we identify drugs that might push the ApoE4 signature to be more ApoE3-like or vice versa? And this is just the pipeline for this. So basically we take a risk signature, and in this case I'm using the NPCs as the example here. And the H9 NPCs were treated with almost 120 compounds. We get this, this huge library of drug perturbations, and then we can compare our risk signatures against the drug perturbation signatures and try and predict drugs that would reverse these risk profiles. And this actually works really well, which is amazing. So here again is this sort of GCP ApoE4 risk profile that I showed earlier. And when this is queried against the H9 NPC drug perturbation data set, we get these drugs predictions where they're predicted to either promote an ApoE3-like NPC state or an ApoE4-like NPC state. And what we have done is actually check whether treatment of this can indeed change the ApoE4 GCP profile. So this is ApoE4 treated with DMSO. This is the ApoE3 isogenic control. And then you can see that ApoE4 treated with resveratrol actually now begins to really resemble the ApoE3 DMSO control in terms of this signature. So suggesting that there's a broad cellular epigenetic change associated with treatment with this drug that might be something really exciting in an Alzheimer's treatment space. We did actually also try the opposite, which is obviously a little bit less therapeutically interesting. And we saw the same thing. We can make ApoE3 cells look like ApoE4 cells by treating with these drugs. This obviously, while not sort of a potential therapeutic, can also start to give us, in the same way that these broad profiles can give us some understanding of what the cellular landscape of risk is, the ability to modify this cellular landscape, to induce it on purpose with drugs, can give us some sense of what are the fundamental pathways, what are the fundamental actors involved in setting up this sort of risk space in the first place. So we can do the same thing with the P100, and this is something that we are actively working on now. So we have this P100 risk signature. Thank you. P100 risk signature that we can query against the astrocyte perturbation drug library, and again get these predictions for drugs that will promote an ApoE3-like astrocyte state and drugs that will promote an ApoE4-like astrocyte state. And what's interesting is that between the NPCs and the astrocytes, the ApoE4 sort of the drugs that would predict an ApoE3-like profile are not necessarily the same across these different cell types. So this suggests that we can learn pretty specifically about like what we may need to look pretty specifically about which cell types are the ones that are most important for correcting. And then we also can learn about the differences in ApoE biology between these different cell types. So it's been a hugely fruitful collaboration project where we've been able to generate these profiles for ApoE. We've been able to, I haven't talked about this yet, I'm going to just talk about this in a second, where we can do some deeper analysis of these profiles. From the sort of original GCP and P100 profiles, we've been able to generate and initially validate some nominated drug opportunities, therapeutic opportunities. And we're now actually exploring even more cell types, ApoE3 and ApoE4 cell types, to expand our understanding here. And then as I mentioned here, I will go very quickly through this as my time is up, but one of the other things that we've been able to do in this space is take this P100 profile, and Karen Christensen will be talking about this tomorrow in session 4 at around the same time, so I highly recommend you attend her talk. But she and her colleagues have developed a means of extracting even greater phosphoproteome data from the P100 data in order to generate, instead of just the 96 profile or peptides, sentinel peptide changes, we can actually now look at 5 to 10K phosphopeptide changes in each of these samples that have already been analyzed. And so this just gives us a lot more biological space to explore, as well as the possibility of now identifying sort of key kinase pathways. So you can see just the number of features she's identified, she and her colleagues have identified, and that this has allowed us to start exploring
We're 2 minutes
potential
over
kinase
time.
pathways. Okay, so I will end there. It's a very tractable model. We're very happy with this collaboration, and there's lots of people to thank in the Scilab, in the Broad, and thank you very much for your attention.
Great, thank you very much, Maeve. We'll go on to the final talk now, please. This is by Duška Vidovic and Mario Medvedovic, where to find and how to use the links, data, and tools from BCIC.
So can you see my slides?
Yes.
Okay, let me just try to turn on my camera too. All right, so hi everyone, and thank you for giving me a chance to present the work that has been done on the in the DCIC 3 centers. In this talk, Mario and I will show where to find and how to use the links, data, and tools. My slides are not changing, sorry.
Try clicking on the slide itself. Oh,
Okay, I got it. So these are parts of the slide that Ajay showed earlier, but actually I would like to start with it as a reminder how diverse the links data is. We also saw that in the presentations by the centers earlier today. So links data spans over different perturbagen types, model systems, and different signature and assay types. And the figure here shows these different reagent and data types as well as the current counts for each of the categories. So with that challenge in mind, the links consortium established the metadata categories and developed standard specifications in order to properly describe and annotate different types of links data and to enable data integration. And here are the two main categories, the reagents and the assay experimental metadata. And their subtypes. And each dot here represents a specification. For example, we have 48 fields to describe a cell line reagent. The metadata standards are released at the linksproject.org, and they're also deposited into the fair sharing as a part of the effort to make the links data fair. So when it comes to the fairness, I will present the main fairness principles and details related to the links data fairness later in the poster session. So after the data and metadata are submitted through different tools developed by the DCIC, or after they are retrieved from the center's repositories, such as HMSDB, Panorama, or Synapse, the data gets validated and then gets processed and packaged into the data sets. Then the data gets QC'd, and then the final step is the release into the links data portal. The data packages can be found in the links data portal under the datasets application, and then from there they can be filtered by different annotations such as assay methods, assay names, center name, project, and so on. And here's one example of a dataset landing page, like one dataset. So the dataset is described with many annotations that can be found under the description tab, such as the dataset name, PIs, funding, assay information, and the list of associated reagents with the corresponding metadata can be found under the metadata tab. And then under the download tab, the data packages are organized by the data levels and can be downloaded. And we also have here the data versioning and citation. So in order to further improve the accessibility of the link signatures, we have also parsed the high-level data from the dataset packages as they are released into the LDP. And after annotating and matching each signature with the link's metadata, the signatures are stored in the signature database. Signatures are organized in a way that they can be individually queried and computed. And also global search is enabled across various reagents and signatures. The signatures are available in the links data portal, and they be queried in a few ways. They can be queried by the metadata. For example, typing in EGF will match signatures with the annotations, with any kind of annotations actually that have EGF string. And here the results are related to the EGFR and EGFP genes. And here in a few sections that are separately, that show actually they're separately annotated as small molecule targets via the mechanism of action annotations, or as the CRISPR reagents with these two specific gene targets. And then we see also the signature types. The signatures can also be queried by genes. For example, for provided list of predefined up and down regulated genes, the concordant and discordant signatures can be found. And this search is enabled via integration with the links, via integration with the iLinks. Each signature is associated with annotations related to the perturbagen, to the model system, and the signature type. And signatures can be downloaded as a subset of associated signatures with the search results, or as an individual signatures. And that's the advantage actually of this new version of the links data portal when the user doesn't have to download the whole data set package. So the user can go straight to the querying the data actually with the searches and getting individual signatures. Another way to query the signatures is by chemical structure, either by a chemical similarity or by a substructure search, as this example here. So the identified small molecules are sorted by relevance, and when selected, for example, here we have lepidolite. For each molecule, a set of annotations can be previewed in the left side panel. So now if we click on the small molecule, then we go to the small molecule landing page where we can find different links metadata, calculated physical chemical properties, and annotations derived from like other sources, such as the mechanism of action and small molecule target. And these particular ones are, for example, coming from Campbell. Recently, we have also set up Google BigQuery, and we're loading more data into it. Basically, we take the signatures from the signature store, validate them, and export as a set of CSV files, and then they're loaded into the BigQuery. So the additional benefits of BigQuery compared to the links data portal is that they enable granular and fast querying and the data access. The BigQuery has standardized metadata and high-level signatures. It's integrated with the links data portal. It represents a persistent, long-term, no-cost access to links data and enables future integration with other BigQuery datasets. The BigQuery can be accessed from the links data portal. So here's the icon for the BigQuery. And here are some examples for the granular queries. If, for example, we search for a specific gene and execute the query, the BigQuery results are returned as the individual endpoints, such as for example, the gene expression for a single gene from a signature that we did a search for. So in this example, so we see the value, like this is a Z-score, this is the L1000 data, and then the measured entity ID is the entry gene of the EGFR gene. So this is my last slide, but before switching to Mario, just to mention that we will cover the links data portal and the BigQuery in the upcoming workshops and the poster sessions today and tomorrow. And thank you for your attention. So let me just stop. I'm still sharing, sorry.
I hope you all can see the next slide now from the same presentation. I will be talking about finding links tools. And I will start by the first place where you go when you are interested about links project, and that's linksproject.org. There is a link on linksproject.org, which there where you can go, and it's going to list all the tools that were created by either links data generation centers or links DCIC, or through interactions between different centers. As Peter already described, one of such collaborative project was development of the GR calculator. That was a close collaboration between DCIC and HMS links project. So this is somewhat overwhelming. So there are tools that can be used to filter these by different types of tools that you may be interested in. Today, I will focus on some of the tools that were actually developed by DCIC. And these are all going to be previews of the workshops that are actually going to happen tomorrow that will dive a little bit deeper into these tools and show you how to use them. So the first tool I will feature is iLinks, which is Integrated Links Data Analysis Portal. This is a user-friendly, interactive data analysis portal that does not require any sort of computing expertise except conceptual understanding of what you're trying to achieve. It integrates a very large set of omics data sets and signatures with analytical tools and interactive visualizations into one coherent analysis platform. In terms of signatures, signatures in iLinks are dominated by Links project generated signatures, L1000 and P100 mostly, but then there are some other signature libraries derived from non-Links resources. In terms of data sets, the data sets are mostly non-Links generated, but also Links data sets are featured, and one can access them and analyze them through iLinks. So the conceptual workflow through iLinks starts with a signature. In iLinks, signature is defined as a profile of changes in gene or protein expression after cell type was perturbed in different ways. Perturbation can be a classical chemical or genetic perturbation, or it could be a disease. So iLinks holds more than 220,000 now such signatures pre-computed. It also, user can submit their own signatures. User-submitted signature can also consist of just a gene list or a gene list of up and down regulated genes. And finally, user can analyze one of these very large number of omics data sets to generate a new signature de novo by selecting a set of perturbation samples and control samples, compare them directly by using iLinks, and then analyze them as a new signature. Once the signature is constructed, there are classical tools that allow for interpretation of these signatures and learning about what they biologically mean by running various pathway enrichment and network analysis. But also what is more interesting probably is that by having these signatures residing within iLinks, you can actually run these additional types of analysis by first connecting your signature through sets of chemical or genetic perturbation signatures, and then try to interpret these results in the context of enrichment or pathway or network analysis. And I will show you an example of how this works. So ultimately, what leads from either starting from a data set or a signature, that you gain biological insight, but also identify putative therapeutic targets and agents that could affect the biology that you started with. So I will kind of demonstrate this in like just a couple of slides, the advantages of having these perturbation signatures to be used in this analysis by analyzing by a canonical analysis of a everolimus signature that we know what it does. It's an mTOR inhibitor, and we picked up this signature directly from iLinks, right? It's pre-computed signature residing there for us to analyze. So if we just submit this transcriptomic everolimus signature to enrichment analysis via Enricher, and look at the pathways that are enriched, we see some biological processes that may or may not be of much interest to us in terms of explaining what does everolimus really do, right? But if we apply this alternative route by first connecting everolimus signature, and here is kind of a cartoonish representation of this signature with only 6 differentially expressed genes. And the first analysis consisted of basically looking for enrichment of such differentially expressed genes in the pathways. If we now instead look at connected consensus gene signatures, these are columns in this little matrix, right? And instead of looking at differentially expressed genes, we look at the genes that were perturbed in the signatures that were correlated with the everolimus signatures. And now run enrichment analysis, the same type of simple enrichment analysis on these signatures. And this is done again by a few clicks through the iLinks user interfaces. Ultimately, we end up with the results that are directly reflective of the everolimus underlying mechanism of action, right? We pick up mTOR signaling pathway as a top target of everolimus. So this is a brief kind of illustration of how this works with genetic perturbation signatures. Similar analysis can be run by looking at known targets of chemical perturbagens. So if we correlate now the same signature with links chemical perturbagen signatures, again, the top correlated signatures are related to mTOR targets, and by analyzing these targets, we could end up with the same result as using genetic perturbation signatures. Different links tools and iLinks among them are in the background connected among themselves, and Dushit's already alluded to this connection between Links Data Portal and iLinks. So both of these resources have full-featured APIs, and basically iLinks leverages comprehensive metadata that is held by Links Data Portal, whereas Data Portal leverages iLinks Analytics to provide sets of connected signatures. And again, there is a workshop tomorrow where we will talk more about the connection between these two and how to use LDP and iLinks in the analysis of your signatures. Another tool that iLinks connects to is GRAIN. This is a platform that continually processes and stores all the RNA-seq data in Geo, and it basically contains all the reprocessed RNA-seq data in Geo, which then you can construct a signature and submit it to iLinks for the analysis, right? And vice versa, iLinks indexes all the data sets processed by GRAIN and reroutes users to GRAIN to analyze them, right, when of interest. iLinks also links out to other resources that help with annotation and understanding of chemical perturbagens or other types of perturbations. And in terms of analytical tools that are leveraged against signature within iLinks, they consist of several iLinks tools, right? These are the ones highlighted, and also tools that are created by the rest of the community. And one of these tools, PyNet, also interacts in both directions with iLinks. There will be another, well, I'll say a little bit more about PyNet.
1 minute warning.
Thank you. So PyNet is this tool that is designed to actually specifically interact or analyze proteomics signatures, which are much more complex to deal with than transcriptomic signatures that tools in iLinks mostly deal with. So what PyNet allows you to do is to input these peptide and PTM profiles. It annotates them and then maps them to pathways and interaction networks, and finally also connects them to other link signatures by, again, API queries to iLinks. And the last, and again, there will be tomorrow a workshop that discusses PyNet specifically. The last set of tools that I will mention are tools developed by Avi Mayan's group, and there are quite a few of them. Enricher, everybody knows about this. Then there is this, for example, large-scale visualization tool that their group develops, and there is this tool that connects basically a fairly diverse set of links tools into a scripted workflows. So I kind of picked up 3 tools. Avi's group has a workshop tomorrow again that will talk more about their tools. I would like to acknowledge everybody in Data Coordination and Integration Center. It's spread across 3 sites, Cincinnati, New York, and Miami. And this is a photo from one of our, actually our last data science symposium that was held in Miami in 2018. Thank you.
Great, thank you very much, Mario. I think at this time we'll try to go to questions. So we've been very fortunate. Many of the speakers have addressed some of the more specific questions privately. So we'll try to just hit some of the big ones. So maybe just to start, Peter, you alluded to the cell system study that talked about reproducibility, and I get the impression that many of our audience members may not be familiar with that study. Can you just in a nutshell, what were the simple suggestions that allowed to improve reproducibility from that study?
Yeah, so let me say what the finding was and then what the suggestion was. So one of the key findings was that the primary sources of variability were not the ones we expected at the outset. So these were done in MCF10A cells, and we had expected that that having multiple different origins for that cell line might be a primary contributor. I mean, we tested that directly and it wasn't. It doesn't mean that isn't a contributor in other settings. And in fact, Todd has written about that in some very interesting papers. And we also found that, variation in reagents, what was not actually the primary contributor, it really was the use of assays that were different, as people perceived assays as being similar to each other, but whether you measure something by counting cells directly or alternatively by measuring the amount of ATP in a lysed cell extract are used interchangeably, and I think we would immediately appreciate that those are not the same thing. A second thing we found was, and this is related to what we just heard from the DCIC, was actually tracking the data from all the way from level 1 through level 4 with known algorithms. In microscopy, that was an issue because a lot of manufacturers provide closed source algorithms, and those actually were not the same either. So one could come up with some fairly concrete suggestions on how to do that. I think what was surprising is the extent to which in practice different centers simply substituted methods for each other if they weren't familiar. So I think that was one of the key things. There were also a whole series of variables had to do with analysis. We locked those down ahead of time, accounting for cell division. Some questions in the chat have asked what happens if cells are treated differently ahead of time, like when you divide them, when you split them. That's all biological variation. So the final thing is, I think it comes down to basically good analytical chemistry. It was not what people wanted to hear. It was 1940s type of science done well. The last thing to say on this is, and this was, I think, the note of caution, is that people were often testing a substitute assay on a very narrow range of conditions, saying, okay, well, let me take one cell line and one drug. This assay is the same as that, or it doesn't matter if I use a different procedure. But over the entirety of a Lynx cube, that makes a huge difference, because what we call context-dependent sensitivity, in fact, is largely a biological variable. And these are the kind of things that had to be controlled for the L1000 data set. Cell density, plating concentration, all of those things were very critical. So those were the type of things. So there was no single bottom line, but we came up with, I think, a series of insights into what was there, why it arose, despite what seemed like good practice, and how it could be solved in the future.
Great, thank you, Peter. I should have mentioned too, NIH has asked us to eat a little bit into our break because we kind of ran a little short. So we're going to take questions until 10 after and then resume at 2:30. The next one's for Shankar, from Shankar to Ravi. Is there any correlation between your expression signatures and gene mutational signatures, both germline and somatic, specifically somatic, including those in regulatory genes?
I don't have an answer as the short answer, but we have all of the information on all the variants, and we are at this stage we're conducting the analysis. So I cannot tell you which of the resolved vectors, if any, correlate with whose expression pattern correlate with the variants. Sorry, but in a few weeks we should, and I will send it to Shankar. And hopefully we'll write a paper soon too.
Great, thank you, Ravi. This is a question sort of to Laura. Laura, you sort of anticipated this in your talk, but maybe you can address it specifically in terms of treating cells with pre-specified mixtures of ligands. Have you done that? Are you looking for it? I think the person in the audience thought that maybe this might be a more biological way to stimulate cells.
Yeah, thanks for the question. I do agree that, you know, in a perfect world with unlimited resources, we would be able to build up to these more complex assays and treatments where we would in fact treat with more complex mixtures in a systematic way. So that's definitely something where we are thinking deeply about. In terms of the logic on this first experiment I described, the idea was to try to come up with a fairly simplified system so that we could lay the groundwork for understanding how do cells respond to these perturbagens that we know something about and we can control as single agents, with the idea that understanding that single agent response will provide the baseline from which we can then perform more complex experiments and think about combining different ligands. We have started down that road a little bit in terms of looking at the phenotypic responses to combinations of some of the ligands I spoke about today. And indeed, what we see is quite interesting, is that in some cases, ligand combinations induce whole new phenotypes. So there's certainly a lot more to explore as we head down that road and thinking about the complexity of a in vivo biological system.
Great, thank you, Laura. And it was a question sort of actually kind of to you and to Peter. So Peter showed with a GR metric, you know, what a useful tool that is for correcting for drug responses. And Laura, you've seen really big effects of cell density on various biologies. And so I wondered kind of to what extent the sort of spatial distribution of the cells, sort of how clumped they are, how distributed they are, what, you know, does the GR metric capture that, or is there still an independent component of variability that we need to somehow measure and correct for?
Well, I can say that something like GR metric was intended to be very simple and essentially, you know, to take 5% extra work to do. You just have to measure initial cell number, basically. There's no question, and there's been some quite nice work from Vito Quarenta and others, that, you know, the more sophisticated the assay, the more you will capture with respect to the type of phenomena that Laura described. However, also specifically with respect to density at the time of treatment, plating density, treatment, what we found in a medium-sized study, so about 60 lines and maybe 50 drugs, was it's significant for about 10% of those pairs. So again, it's one of those things that I think is revealed by large-scale studies, is both the negative space is very informative because a systematic analysis, as Todd described, tells you both where there are signals and there aren't signals. So the flip side is to say, you know, for more than 80% of cases, cell density is a fairly small contributor. For the remaining cases, it's a dramatic contributor. And for a drug effect or a CRISPR, it can either increase sensitivity or decrease sensitivity. And you can, in some cases, we never chase those down completely, but you could rationalize them in terms of typically cell non-autonomous autocrine type effects.
Thanks, Peter. I agree with all that Peter said, and I think one of the key things is making the community aware of these different issues, so that as people are running their experiments, they understand how to run them in a controlled fashion, and also what we should report. You know, Lynx has spent a lot of time thinking about what are the metadata and key variables to report in one's experiment. Some I think are you know, well appreciated by the community, but what some of these studies have uncovered are additional variables that are important to report out, so that we can think about how to reproduce and how to compare across results from different labs.
Great, thank you, Laura. Hey, Todd, Jessica Ewald asks, much higher level biological interpretation of transcriptomic data relies on gene set pathway analysis. How do you do this with a panel of 1,000 genes where you likely have only a handful of genes per pathway? And can pathway perturbation be inferred in some way?
Yeah, that's a good and common question. I think that you know, some of the effort that we do is to try to reconstruct the rest of the transcriptome that's not measured by inference. I would say that works okay. It's not spectacular in its accuracy, so you can pick up some there. I think the other thing is that in some cases it's possible to establish connectivity between cellular states, even just with 1,000 transcripts, but not necessarily being able to put a label on it in, you know, a typical pathway type, you know, nomenclature. I think it does raise also the interesting question, you know, to what extent do we really believe that the named pathways are really the gold standard, or should they be reconstructed and relabeled based more on empirical evidence and more data-driven rather than, you know, a biological understanding basis, which is the current situation.
Okay, thanks, Todd. Clive, there's a question sort of, you know, just the general sort of philosophy or approach to assessing the validity of iPS models. The starting point clearly is a genetically precise cell model of a patient, but, you know, some people have raised concerns about aging for certain diseases, and there can be a trade-off between the simplicity or complexity of a model and the ease, throughput, and reproducibility of the model for evaluating perturbagens. Kind of what's the thinking, what's your thinking or Neuralink's thinking about that?
I think I use the V word, which is validation. I think for all of us, if you're going to validate any of these hits in any of these models across into patients, that's when the rubber really hits the road. I think all of us are in that position, you know, for iPS modeling, of how far do we have to go in our modeling, or how mature do we have to make the cell to make it valuable in terms of hits and pathways. And the only way I think to answer that is to do some very direct correlations with what we're seeing in the Lynx program and clinical patients, right? So does your drug, you predicted it would do something, and now do a trial to prove it. And I think that's where pharma will get more interested, because there really is a value to what we're doing. And I think the validation projects should be where Lynx goes next, to be frank. And then you'll get the value of these models. Whether the context is important because we grow cells that are much more like the brain, or whether you could just do it in a MCF cell or a cancer line, it's a big question. I mean, if you could do it in a cancer line, don't bother making iPS if you get the same fidelity of data. But if you don't, then we're going to need to do the iPS models. I'll open that up. That's more of a discussion point to open up, Steve. But
thanks, Clive. Yeah. Maeve, there were a couple of questions kind of related, so I'm going to kind of mash them up. Sort of, I think the general question was try to understand the signatures that you saw and to put them in context. And so I think one of them was just, to what extent have you looked at other cell lines, and have you seen a correlation of the histone signatures against ATAC-seq data on the same cells?
So for the first question, we have definitely looked at, so the two independent generated isogenic lines. We looked at one generated from an ApoE3 patient and CRISPR edited to ApoE4, and one generated from the ApoE4 patient and CRISPR edited back to ApoE3. So we feel pretty confident. The only, when we are considering what to sort of look for in a signature. We only looked for things that where the change was in the same direction between both parental lines. So hopefully kind of showing us something that is ApoE4, more an ApoE4 versus ApoE3 specific effect versus some of the other things that might be going on in the genetic background of any one particular individual. But as I mentioned in the text answer to this question, I think that, you know, adding more as we're seeing, you know, as more and more IPS lines are established, that, you know, it only increases our confidence to look at even more isogenic lines, or if isogenic lines are not available, then like just larger panels of E4 versus E3 and see how broad the effect is. So we're pretty confident because we have the two well-matched lines, but and we only looked at ones that were the case in both, but it's always valuable to look across a greater number of independent cell lines. To the ataxia question, we have not actually looked at that yet, but we are interested. We're starting to kind of scale up to do some ataxias on all our ApoE lines. So we will hopefully have this kind of information for our ApoE various cell types soon, and we can correlate, but as to yet, we don't know. So we just have sort of the GCP profile, and we're working off of that.
Great, thank you, Maeve. I'm not sure if this question is best to Mario Dusica or Todd, but the question is it possible to download not just the perturbation states of 1,000 landmark genes, but also the inferred 10,000 genes that are used for connectivity map scoring?
I'm happy to take that. Yes, you can get those inferred transcriptomes at clue.io. I'm actually not sure if they're also available from the DCIC, but you can get them from the Clue website.
Great, thank you, Todd.
Yes, they are available from DCIC as well.
Okay, perfect. And Mario, there was another question. There's a little confusion about iLink versus the links portal. Could you just clarify that?
Right, so kind of bird's eye view of these two would be that links data portal is about data access and meaningful ability to kind of navigate the links data, and iLinks is about the data analysis, and they're connected.
Great. Another question, Todd, the slide you showed about genetic versus chemical perturbation was fascinating. You know, it looks like some small molecules really reproduce a genetic ablation and others are very different. A lot of pharma now is really being driven by genetic links between a target and a drug. I guess the question is, do you have any advice for them? Because it doesn't sound like it's necessarily a straight shot in terms of developing small molecules. Is there a way, you know, what would the approach be, I guess?
Yeah, so the approach that we're taking, for example, when a therapeutic target is suggested from a genetic perturbation screen, for example, a genome-wide CRISPR knockout screen, that not to rush to a drug discovery strategy based on, you know, maybe the most obvious enzymatic activity or domain of the protein, but to do some careful mapping to ensure that that strategy recapitulates the genetic finding, because it may not.
Great. Peter, Dilla at UTM wanted you to talk a little bit more about the Alzheimer's study that you mentioned. I think she specifically wondered about the logic behind focusing on drugs that have positive connectivity score to disease signature.
Yeah, so the paper, the method is called Dryad, D-R-A-I-D, and it's out in bioRxiv. I can try to post it in the chat in a quick second. The idea, it was essentially a proof of principle. So what you find when you do, I think we all know, when you do a signature based on a gene set of a perturbation or disease process, and you get a, let's say a highly predictive signature, even if it's statistically significant, what you find is there are multiple replaceable versions of that. So there are different gene sets that can be used and be equivalently predictive, even if a random one doesn't. So the goal was to see if we could focus predictive gene sets on things which drugs perturbed, and ideally FDA approved drugs perturbed. So that was the idea. It was essentially a form of feature selection for modeling, for a machine learned model or a statistical model based on a drug-based perturbation. Now, in the end, you can't specify the sign in that because you can end up with things that are either pro-Alzheimeric or anti-Alzheimer's, and so you have to then do a follow-up experiment to actually work out. We knew in the cases to which I briefly alluded, and you'll see in the bioRxiv paper, we knew that those were anti-inflammatory, and there's quite a bit of evidence that JAK-STAT inflammatory mechanisms are involved in at least some sets of Alzheimer's. So essentially, the logic was a feature selection logic.
Great, thank you, Peter. Back to Mario and the DCIC, Amrit Singh asks, is there a way to access specific versions of LDP APIs in order to ensure reproducible workflows?
Yes, that would be the simple answer. Yes, the LDP has full-featured API, so that's possible.
Yes, so I can just add, actually, we version the data sets. So for the user, actually, through the API, the user can get the latest version of the data set. So, and it can integrate and have reproducible workflows. But we also track the previous versions of the data set. So just keep in mind that the data sets sometimes change.
Sure.
But the workflows are constant. The API is there.
Great, thank you so much. So we've run out of time, it's 11:10. I think the plan is to resume at, or 11:10 Pacific time, to resume at 2:30, so in 20 minutes. And I want to thank all the speakers for a terrific session and the audience for great questions. Thank you all very much.
Thank you, Steve, for herding cats.
Meow, meow.
Thank you.
Thanks.